Good morning, everyone. We are going to start now. Welcome to the first session of the second day of IRBLC Master Thesis in Thesis. My name is Gosechan. I'm from University of Western Brittany in West, and I will belong to the different members of the jury. So we first have Barbara Cantinai, professor here at UniVPM. Next you have Nico Weidberg, who is a professor as well at the University of Oviedo in Spain, and Nicolas Jegli, who's a postdoctoral researcher at the University of West. So we'll start with Dylan. Dylan, so you have 15 minutes. Whenever it will only 2 minutes will remain, I will wave at you or make a sign to let you know that there's only 2 minutes and then to stop completely.
Okay, so good morning, everyone. I'm going to talk about my thesis project on the behavior and physiological responses of the invasive species Amotea hilgendorfi to thermostressors. So to start with, to give a brief introduction, invasive alien species are known to create ecological disruptions, and these disruptions can be exacerbated by climate change. There are approximately 37,000 invasive alien species known, from which about 10% have known to have a negative impact. From this, in the North Sea, we know that there are 167 species. And from these species, there is an overlooked group called Pycnogonida, that is a class of Chelicerata. And they are not actual spiders, they are the sister group of Euchelicerata. But in this Euchelicerata is where land spiders are. Amotea hilgendorfi is the first known pycnogonid to be an invasive species. We know that this species has actually one of the highest abundances in the world for this group. In addition, we know that this species can have multiple mates, and they have an ectoparasitic larvae that parasites hydroids, and also because of the genus, we can say that it can be a generalist feeder, however, we don't know. In addition, we know that these species can be in a very wide distribution in their native and invasive habitats. This is a timeline of the invasion of the species, and we can see that the first record was in 1978 in England, and now we have the highest abundances in Belgium, about 1,000 species, 1,000 individuals, sorry, and we can see that now they are also in the Mediterranean Sea. There are about 6 countries that are involved, but of course, it's very sure that they are in so many other countries. The research objectives of this study was to experimentally assess how the thermal stress influences the behavior and physiology of the species. We wanted to compare the temperatures 15 versus 25 degrees with the idea of simulating the two temperatures of the native and the invasive areas. We had 3 main questions. The first one was if there is an effect of the elevated temperature in locomotor performance. The second, if there is an influence of temperature in the habitat selection. And the third one, what is the upper physiological thermal limits of the species. In addition, we wanted to know what is the success, if this success is supported by the behavior and the physiological plasticity of the species. We want to establish a data set for future comparison with the native populations. We did this study in Knokke in Belgium. We sampled the invasive oyster over these wave breakers. Then we take the animals to the lab and we basically extracted them, sorted them, and sexed them. Then we put them into acclimatized tanks at 24 degrees and 15 degrees. Then these animals, to do the first experiment on the behavioral trials, we basically put them in 2 different temperatures, 15 and 25, and we had 12 replicates per temperature treatment. Then these replicates, we basically recorded them and we see how they move. And in addition to this, we added like a light cycle to see if there is an influence on the photoperiodic behavior of the species. In addition to this setup that I explained before, we had the first experiment that was basically a flat tank that was empty, that we call blank. Then we had the inclination one, where we had an inclination of approximately 60 degrees. And it's exactly as the previous one, and we wanted to create a gradient of shallow versus deep zones. And then in the third one, we added the substrate that we found in the habitat where we sampled them. Then for the physiological trials, we put the same animals that we have nematized before, and these animals we put to see where is the time when they start dying, before they die. So we manipulated them to see when they stop dying, and then we recorded this time. Then for the data analysis, we did the manual tracking of the videos that we recorded for 24 hours for these 74 replicates, which was the hardest part to do. Then to this, we created an R script to know the total distance, the velocity, the percent active time, and the spatial activity of these animals. Then we did no parametric test to test for significance. And then for the physiological data, we had the median knockdown times that I mentioned before. And with these median knockdown times, we created a model called TDT that I explained more at the end. And then we did an ANCOVA to search for also significance. Our results show that we sampled 52 oysters, from which we extracted 191 individuals. And this is the highest ever recorded abundance of the species. That is about 3.7 individuals per oyster, which is higher to a previous study. And this can mention that maybe there is already an advanced invasion of the species. Also, we think that maybe this is the main predator over the shells. In addition, we know that because of a previous study made by my supervisor, they found that approximately 97% of the community structure is just this species. So we can see that basically the native species disappeared, and this can be also compared with a study that was done in the Venice Lagoon. These very high abundances can be explained by an effect called the boom and bust that occur in very early stages of an invasion, but it can be also an impact lag on the impacts of this invasion. For the experiment that I mentioned before, the tank that was basically flat and with anything on it, we can see that the animals are very active during the day, are very active in 15 degrees. However, in 25 degrees, they reduce their activity, the percent of time that they are active. And this is also shown by the median velocity and the total distance that we can see on the graphs. Something very relevant is that these animals are on average like 1 centimeter, 1.5. And if you can see here, they can walk up to 63 meters, which is something very relevant. These animals, they don't have a pelagic larvae. So of course, the movement they can do as an adult is very, very important. And of course, this can explain the rapid spread of the species. We can see in these cumulative distance travel plots that over time there is a continuous movement. However, if we see like the inclination of this movement, it can be reduced during the night. And this is also seen for the velocity profiles. Then for the heat maps that I created for the occupancy, we can see that the animals have a very interesting behavior that has been seen in different animals. That is called taxis, that is basically an edge-following behavior that can be explained by a shelter-seeking behavior. Maybe they want to explore the area, they are stressed as well, and this movement also can explain aggregation and normal movement of the animals. Every dot represents the occupancy of the animal over time. Then, for these experiments, I try to see if there are significant differences between the treatments. So we can see that the total distance traveled by temperature has a significant decrease in the higher temperature, and this is also seen for the median velocity by temperature. So we can say that there is a significant suppression of locomotion at higher temperatures. In addition, we can see that the photoperiod behavior, the animals tend to move more than in the day than during the night. Then for the inclination experiment that I did that I explained before, we can see that the animals also have a similar pattern. They are more active during in the 15 degrees treatment than in the 25 treatment. And something relevant here is that we can see that the animals have a strong preference for the shallower zone. And something also very interesting that was just by coincidence, one of the times I added less water, and I saw that the animal was able to go outside of the water and stay there for 17 hours and then come back and keep moving. In this experiment is where I saw the longest distance travel, that is for about 81 meters over 24 hours. A similar pattern that before we can see in the inclination distance travel for 15 degrees is continuous and is slightly reduced during the night. However, when we increase the temperature, we can see that during the day, the animal is moving less, and during the night is moving more. We can see exactly the same for the velocity profiles. In our heat maps, we can see that the animal has the same behavior, that is walking on mainly on the corners of the boxes, and is spending most of its time in the shallower area. Something very relevant is that we didn't see any, in the overall locomotion activity, no significance between both temperature treatments. However, we can say that there is a strong preference for the shallower water areas, that biologically this can explain that in these shallower areas they have more food availability. The hydrodynamics of the environment is also interesting for them, and also how the light penetrates is completely different. The highest point, the crucial point on this experiment is that we can see that the animals can switch their behavior and stop being diurnal and start being more nocturnal. So this means that the animal can select shifting when active but not where in the vertical areas. Then, for the last experiment that I did for the behavior was when I added the substrate. I saw that the animals completely stopped their movement. Not completely, but they reduced it 50% compared to the blank experiment where I didn't add anything at all. And then we can see as well that the animals in both temperature treatments have exactly the same activity. So we think that this can be give us an idea that maybe the animal is trying to hide from the light. In addition, we can see that the animals have a strong preference from the oyster, which makes sense because this is where we started sampling them. Also, it's interesting that these animals are known to be cryptic. That's why it's very hard to find them. And of course, this experiment can show us something interesting as well is that I always saw them going under the substrate, so they try to be hidden. Now, something similar pattern we can see here is that during the night it looks like they are slightly more active for both temperature treatments. And this can give us a slight idea. However, I didn't check specifically for this, but in the lab I'm working on, we already did some experiments and we know that they don't like the light. It's not included in this experiment, but I can just explain it now. Now if you see, like these heat maps has like a lot less dots. This means that they are moving less. And we can see something interesting is that they are always choosing the oysters. And when I did this test for both temperature treatments, we can see that there is no significance, as I mentioned before, in the overall locomotor performance. However, we can see as well, as we mentioned before, that there are no significant differences between the day and night. There is something very relevant, is that when we start increasing the temperature, the animals, and this is something that I have it in this plot, start choosing more muscles. So this can have a strong explanation that if you think about it, they will start invasive. If the temperatures increase, they can maybe start using the muscle beds as a substrate. And this is where you can see the muscle, from muscle to blank, they basically start using more. Oysters were still the most used one, but we can see that muscles start being also relevant. Now for the physiological thermal tolerance experiment, we wanted to see if there is a difference between the two temperature treatments when we acclimatize them. So here we have time, and here we have the different temperatures to expose them. And with this model, what we can see is that the animals start dying faster when you expose them to higher temperatures. And with this, what I can do is extrapolate this information to understand what's the highest temperature they can survive, and also what's the highest temperature they can survive up to 60 minutes and then for 1 week. I wanted to check if there is a difference between the demographic groups, male, females, and juveniles. However, I didn't see any significant difference. Also for the acclimatation, I forgot to say it. And something relevant is that the animals can survive for 7 days at 33.5 degrees. And if we put them at 60 minutes, they will survive at around 36.5 degrees. So, of course, this is mentioning that this animal has a pre-adapted invasion, a pre-adapted behavior for invasion, and also a strong physiology plasticity. And this is, of course, relevant. Why? Because it's almost impossible nowadays that the North Atlantic is going to reach these temperatures. And something relevant as well is that maybe even the native distribution of the species can be even going to the southern part of the Pacific Ocean, where this species is native from. So now the conclusion is that we know that the success of this species is mainly because of the synergetic interplay of two main things, the behavior plasticity and the physiological thermal tolerance that I mentioned before. And we can see as a key traits that the adult has a very relevant dispersal capability. There are some adaptive behavioral responses that makes, of course, the animal very resistant to thermal stress. And we know, in general, overall, that the animal has a very strong physiological resilience. This attributes to the resilience and adaptability of the animals. And in addition, we know that there are very strong impacts that we can measure now. Something that I would like to propose as part of this management of the species is that very probably we can use this animal as a model organism potential for overall for the pycnogonids, because we don't know about their ecology, and in addition it's very easy to keep them alive on the lab up to 2 months without feeding them. There are several things that I would like to explore in the future, but because of limited of time I won't talk about it, and maybe we can discuss it in the next minutes. Thank you.
Thank you very much, Dylan. The perfect respect of the log time, 14 minutes and 59 seconds. Very impressive. Has anyone in the jury some questions for Dylan? Who wants to start? Yes.
Okay. Compliments for your presentation. It's very interesting. I was wondering when you were talking about the native habitat of the species, but then during your speech you answered to me, because late in the speech you told that is from the Pacific Ocean. Is it correct?
Yeah, they are from Japan and California, and actually the same experiments, my supervisor already went to do them in California, and he's going next week to Japan to have like this exactly same experiments, but in the native populations.
I'm wondering why it's so important to address and study the locomotion response. Is it connected to the way of introduction, you think?
It's mainly, there are like two main things. We don't know why, in general, for sea spiders, we don't know why they are everywhere if they don't have a pelagic larvae. So probably because of these results, we know that they have a very high capability of moving very far. The second part is that, of course, because if it's an invasive species, how far they can reach is very relevant. So we can know that basically they can be everywhere right now.
Because this kind of species often move all over the world, thanks to Forasia, or so because they live attached to fawning and so on. I think that could, so I think it could be the main way of introduction. But again, nobody knows about the way of introduction.
Yeah, exactly. And I want to show you something. There is a hypothesis that one researcher saying that most likely they came with the oysters. This is a picture of the individuals. And when I saw this, I was like, they literally look like they live here. When my supervisor went to California to search for them, he wasn't able to find them on oysters. They were actually living in different animals like sponges, and I don't remember exactly. I hope if he's listening, maybe he can send a message on the chat. But they live in a completely different habitat. I don't remember exactly the paper, but we think that maybe they come with the oyster. It's just a hypothesis, and that's the thing, that he's going to go to Japan where this oyster is to see maybe they come from there.
Thank you.
You're welcome.
Okay, very nice presentation. I really enjoyed that. Very nice pictures, a nice combination of pictures and photos and diagrams. Anyway, I would expect more of that in results. You put some tables there that would have been bar graphs. You put some bar graphs for some results, and for other, you just put tables that could have been paragraphs. Why this difference?
What, like for the experiments, what I did, like our results, for example, here is just a table just to show like the animals.
But
Then for this,
no,
what you mean?
These are paragraphs, yes. This one, for example, the previous one.
This one.
Could have been shown in a paragraph, right?
Yeah, I think it would make it easier. It's just because I literally tried to make it, I think this, I didn't think about it, to be honest, because this was so much to put into it. I tried to summarize it, and maybe most of my time was trying to summarize. This was so big, like if you see the manuscript, it's huge, because of course I wanted to give as much as detail as possible, so. But yeah, it's a good.
Yes, but in our recommendation, if you have two ways of showing something, especially in a presentation, show the most visual one, which is
The graph. Thank you for this comment.
Then, can you tell me a little bit more about the experimental design? What kind of analysis specifically was that? Analysis of variance, I guess, right?
Yeah, we wanted to see if there were like differences between the temperature treatments, mainly. That's it. So that's why we did the Wilcoxon rank.
But how many factors do you have?
Just temperature. And then like I look specifically for temperature, and separately I just look for
But it
photoperiodic.
was the same experiment.
It was the same experiment.
That's a little bit weird. You have to be careful with that. Because you may have that's kind of analysis that should have been a factorial analysis crossed with two factors. That would be a slope and temperature. And you should have also the interactions in between that, like more slope and more temperature, flat and colder, flat and more temperature, you know what I mean?
Okay, yeah.
So you can see the effects.
Okay.
But you have to reflect a little bit on that. And also about the slope, about the
inclination.
No? I understand the reasoning, but you have an angle in just a short distance. That's not natural, right?
Yeah.
That's a little slope.
Yeah, it's huge.
It's huge. It goes for meters and meters.
Yeah.
So it's a little bit unrealistic. Did you think in that a little bit?
Yeah, like the idea, of course, was just to create this gradient, and of course, just by the results, we can see it. And also, like in the movement, just this slight increase for such a small animal, we think was relevant. That's why we chose this. Of course, also the limitations of having bigger tanks would make it even more difficult.
Yeah, that's true. But also, on the other hand, I think you have to address that this behavior of the animals moving around the wall is because there is a wall. It's because there's a boundary effect artificial that maybe.
Yeah. It's part of our limitations. That is also something I discussed. I didn't mention, but yeah.
And I think there are plenty of species that they don't have pelagic, they have a lot of mobility diagrams, and the juveniles have a lot of mobility, and not only walking, but also just resuspending themselves in the water column, especially the little ones.
Yeah, the little ones.
And going around with the currents that could disperse the animal even farther away than just walking.
There is one register of one species, actually, of pycnogonids. I don't remember the species, but because we know that they are parasites of hydroids, so maybe they use them once they do this metamorphosis into being like a jellyfish. Maybe they can move on the jellyfish, but it's something we don't know. It's of course something I'm just saying by the things I've been reading, but no one proved this before.
A lot of things that are unknown.
Yeah.
Okay, thank you very much and congratulations.
Thank you.
Thank you, Nico. Just to your supervisor insert, it was on sponges and also on acids.
Acidius. I know he will be there.
Also, Nico, if you want to participate,
Yeah, sure. Thank you for your presentation. I guess, I've got a few questions. One is quite general in terms of the conclusion of the study. So one of the conclusions is that you have quite a good mobility of the adults.
Yeah.
And I was wondering whether that reflects. I was wondering if your experiment is a good test of that, given that basically you see a lot of movement where there is no shelter.
Yeah.
But as soon as you put the shelter in there, they, from what they understood, they hide and they
stop
don't
moving.
move that much anymore.
Definitely.
But in the field, they would have shelter pretty much everywhere. No?
Yeah. Yeah, we found them. Of course, this is something that we also start thinking. This is just mentioning the capability of locomotion. We don't know if this is actually what's happening. This is something that I wanted to discuss at the very end. That maybe we require like field validation of this information, because yeah, this was made on a lab just to see how they move, but we don't actually know what's happening in real life. So yeah, I agree with your comment.
I guess I have another small one, which is that might be more the documentation and all that. But I think you said that temperature, water temperature of 35 degrees in the Atlantic are expected in the future. And I'm like, I mean, it's gonna get warmer, but not necessarily that warmer. So
Can you repeat the?
you mentioned that this kind of temperature range for in the Atlantic will be expected in the.
No, I said that this is the temperature that they can survive.
Yes.
Like up to 7 days, like 33 degrees, and I say that it's very hard that the North Atlantic can reach this temperature. So this means that for the egg, it's completely fine. However, something that I found very relevant, and when we went sampling, there was snow on the beach when we went sampling. So maybe the interesting part is to see the other way around, maybe lower temperatures, start testing lower temperatures.
Okay.
That's what I see more relevant because, yeah.
Yeah, no, but yeah, the one relevance that we see for those temperatures though is that they are intertidal, right?
Sorry?
They are intertidal animals.
Yeah, they're intertidal animals.
Yeah.
But, however, there is one record of the species in Belgium, actually.
Yeah.
That is, I think, 30 kilometers away from the coast. And they are
So
about
maybe
30 meters, 30 meters deep. So they probably are everywhere.
No, because yeah, I was thinking maybe you get those kind of temperature in some microhabitats sometimes. But
Yeah.
Yeah. Do you have some? One question. One question. Okay. I also have one. I guess question remark big time on the stats you did.
Yeah.
Because you have a design where you do a lot of essentially pairwise tests between different parameters. And you have tested different things. Do you see an issue with having a number of pairwise tests like that on different parameters?
To be honest, these type of things, I'm not the expert to say. I try to use mainly from my classes. I remember what I did this ANCOVA is, how is it called, a nested test, because we have the temperature treatments, and then we have...
I think that might be fine. I'm more worried about the number of non-parametric tests you use.
Yeah, for this, what did you say before, right?
Similar. It's related.
Yeah, maybe you can explain, because I don't understand. So maybe
the issue when you test, you compare different things, and if you do only pairwise tests, you are pretty much bound to at some point find something significant. Because I mean, with an alpha level at 0.05, which is what we take, it's still a 1 out of 20 chance of finding something by chance. So if you do a lot of tests, you're gonna find something significant. So generally, you want a global test before getting into the.
Okay. And for example, because of course this is something we would like to publish.
Yeah.
So, what could be like from your expertise?
Another Cruise before.
Okay.
Thank you very much. Your small, little last question was not so little, so
Sorry.
Congratulations, Dylan.
Thank you.
Hi everyone, my name is Juk Valiance and today I'll be talking about environmental drivers of benthic biodiversity in an Antarctic fjord named Collins Bay. First of all, I want to introduce the Antarctic food web a little bit. It's mostly based around krill, which you can see here in the center. And that is transport from the plankton to the bigger animals like penguins and whales. However, the most species-rich part is the benthos. This is every species that lives in or on top of the substrate. And it includes the filter feeders, omnivores, macroalgae. And it's quite important for sequestering carbon into the seafloor. And that's also why I think they're very important. But you have to remember that Antarctica is a massive ice sheet. So this has a very large effect on everything that lives on the Atlantic seafloor. Think about ice scouring, turbidity, meltwater changes, and all of this will change the way a community will be structured. So I wanted to look into this. How is this affecting the benthos? And to do that, I went to the western Antarctic peninsula of Antarctica. This is the most northern tip of the continent, and it's also the fastest heating region in the world. So if you zoom in a little bit, on top there's this little island called King George Island. This is the most visited place in Antarctica. There's a lot of scientific bases, Peruvian, Russian, Chinese, Chilean. And if there's so many people here, how come we know nothing of Collins Bay? It's next to the Collins Glacier, but there's never really been done a complete biodiversity assessment of the species that live here. There's 3 studies that incorporated it into their studies, but they only did 1 sampling in the area or only looked at the first 30 meters of the water. So that led me to my research question. Do a baseline study of Collins Bay benthic community and look at the influence of depth and glacial proximity on diversity indexes, community composition and functional biology. My hypothesis is that the closer you get to the glacier, the more disturbances there are, so there's going to be a lot more mobile species that can move out of the way if a big ice block comes on top of them. And then further from the glacier, there'd be more sessile species, and then there would also be an increase in species richness because of this. Then for depth, I would think that in shallow waters there's more grazers because there's more macroalgae that can grow in the light. And then the deeper water is changed to filter feeders and a higher species richness. And both of these are going to be continuous rather than in bandwidths. So a little bit more about Collins Bay. I had to hand draw this because there's no bathymetry accessible on this area. But just to give you an overview in the area, there's a gray sill in the opening, and then there's 2 deeper areas close to the glacier and 1 in the center of the bathymetry of the bay. Now, this allowed me to do completely cross-design with distance of the glacier and depth. So I placed these stations in this way and was able to cover the entire distance I wanted. Please note station A4, this one is the furthest from the glacier. In this area I did more than 4 samples to get the entire depth and study the effective depth within a small relative place. Now, what I did in every place is an oceanography test, so I used CTD. This allowed me to give the water properties throughout the entire water column and most importantly the water properties at the bottom. Then in all the stations I also looked at biodiversity, so we deployed this mini ROV that was able to film the benthic. What we did is that when it's lowered in the station, it would follow a butterfly pattern around the station, moving forwards, tilting down, taking a moving picture, tilting upwards and forwards again until at least 35 samples were taken or the butterfly pattern was completed. Then from these pictures I was able to deduce which species lived in these areas. It's just an example, but by counting them I was able to create a biodiversity table, and from this table I was able to calculate the total abundance, species richness and biodiversity index for each replicate within a station. And with this data I could understand the effects of depth and distance doing a 2-way ANOVA, I could look at the drivers of a community doing a CCA, and I could look at the community composition combining the functional traits for each species that I found with the data. Now my results. I counted over 14,000 individuals. Most of these were Nemertida, polychaetes, but also a lot of other fauna like sea spiders, icefish, sea stars, ascidians, and then I also counted 1,900 seaweeds. These were done in a present-absence manner because, as you might have seen on the picture, it's almost impossible to count individuals of algae from a moving picture. And then let's go to 4A. This is the station where I did multiple samples in the same area. And as you can see maybe from the pictures, which are just random pictures from the depths, there's a gradual change in the community the deeper we went. And in the table you can also see that the deeper you go, the more complex the community became. And there was no zonation found, it was really gradual. Then for algae we found a reverse effect, so shallow waters had more complexity, and the deeper we went, the less there were, and the less complex the community composition became. But now let's look at the entire place and take the biodiversity tables into account and look at the effect of glacier on these. So here on the left is the closest to the glacier, on the right is the furthest away from the glacier. And you can see that there's a shift from bivalvia and polychaetes to Nemertea and Echidnomata. We found a lot more species than earlier studies have done in this area. That is probably due to the higher resolution, but also because we used the imaging, which allowed us to see a lot more species at once. And this became especially important for the seaweeds. We found 3 times the amount of seaweeds as Valdivia, and 2 times the amount of species as the study did in 2011. Here we also see that no matter where we were from the glacier, there was always a decrease in complexity with depth. Now the biodiversity index is again for the entire area. This is the furthest from the glacier. It's a very clear pattern that the deeper you go, the more complex it actually got in total abundance, species richness, and also Simpson diversity index. However, when you get to closer to the glacier, this effect completely reversed. And you can see that this is a gradual decline. And what is most interesting is that at the shallow waters close to the glacier, where the ice is hitting all the animals right as they are, we found high biodiversity. We found a lot of mollusks, which are pioneer species. That is similar to what is found in Marion and Potter Cove. We found Echidnomata, which are highly mobile, so that was also kind of expected. But we also found a lot of ascidians, which are filter feeders, sessile animals that cannot move away from any ice. So how is this possible? Why are they in this area? Well, we hypothesize that it's because of pelagic benthic coupling. So even though the Antarctic waters are very mixed, there's still an extra inflow of nutrients from the ice water that melts into it. So this could increase the chlorophyll A in the water and therefore increase the amount of filter feeders. However, to be certain of this, we would need to do further studies, and I can't really prove that this is the reasoning. Then, for the seaweeds on the right, there was expected results that the deeper you go, the less species there are, but there's no clear trend considering the distance from the glacier. It just becomes a little bit less complex the closer to the glacier you get. What is interesting is that we did not find any pioneer species close to the glacier. So even though we found these for the fauna, we did not find them for the seaweeds. This was really unexpected, because especially Palmaria decipiens, we can find that all over the Western Peninsula close to the glacier. So this would mean that there is a local shift in Collins Glacier, that even though they're close to other places where we find this plant, it is different from what was expected. And then between minus 20 and minus 40 meters, we also found a lot of diatoms in the form of colonial gnats. And this is quite interesting, because a diatom, especially in these amounts of blooms, are an indicator of ecosystem change. So the fact that we now have a baseline of what is happening in Collins Bay means that in the future we can see what will happen then. Then last but not least, the functional biology of the entire bay. So again, close to the glacier we found very low mobility. This was also unexpected, because normally it's the reverse. We found a lot of filter feeders in this area. It might be because of self-recruitment. In the deeper area, close to the glacier, there's a sill not connecting it to the other part of the deeper areas of the bay. So there might be self-recruitment in this area. And also, most of Collins Bay is very muddy. There's very little hard substrate, especially at the depths. And what could happen is that there's a filter feeder that attaches on the small area of the rocks. It might increase the surface and therefore increase the biodiversity locally, which is another proof for the patchiness that we found. Then, the further you go from the glacier, there's an increase in mobility and an increase in functional group diversity, similar to Marion Cove. Then the feeding mode proportions. So on the left, again, closest to the glacier, on the right, furthest from the glacier. On the left, it's mostly filter feeders, some suspension feeders, and this gets more complex the further we go away from the glacier. Suddenly there's grazers, opportunists, deposit feeders, And this is a very interesting baseline, but it can't really compare it to other studies because there haven't been studies on this specifically in Antarctica as of today. So that's something for the future. Then my conclusion is that the benthic biodiversity in Collins Bay is strongly influenced by the effects of depth and distance from the glacier. There's a high species richness even in shallow waters, which is quite unexpected and interesting for the future. And then fine-scale monitoring is going to be needed for the future to understand any changes that might come with climate change. Then it has been sent in for publishing. If you scan this QR code, you can find the preprint online and also some extra pictures of the project that I could not post in this presentation. There's also my references in there. And then I want to thank especially Ro and Nati for steering the boat in the cold weather, and Antonia for steering the ROV, and Cesar for making all of this possible. And a thank you to you.
Thank you very much. We have plenty of time for questions. Who wants to start?
Okay, thank you very much for the presentation. I enjoyed it a lot. But just a curiosity, I wonder if you also think about the presence also of the movement of the sea ice or the iceberg. There is also a scar, the ice scar, that could be important in that area.
Yeah, the ice scouring, you mean?
Yeah.
Yes. So,
Because you say that there are a lot of sessile species, and so probably close to the ice there is no this effect, could be? That there is no scar effect?
well, that could be, but we did see a lot of ice fall into the water as we were working there, so I don't really see that that would be the reasoning. Also, if you look at Marion Cove, which is adjacent to this bay, you can actually see a lot of scarring from the ice. So it was really unexpected that that wouldn't be here, but it's very interesting, yeah, and unexpected.
And that's another curiosity because I know that sponges are really important in the benthic community in Antarctica, but if I understood well, there are not so many in this area, or are they numerous?
There are some. Most of the species close to the glacier that were filter feeders were ascidians. There were some sponges that found further away from the glacier, so that would indicate a further successional stage. Yeah.
So you find also sponges that are big, and so they are, it means that they are in the final stage of succession.
I didn't find the massive sponges, but I did find quite some different species of sponges, yeah.
Okay, well, first of all, congratulations for a very nice presentation. I really enjoyed it, especially the way you were addressing the public and explaining everything, pretty clear, pretty neat. I really like that. And the presentation is very nice with linking the different parts of the ecosystem with nice photos, nice figures. But I wanted to get a little bit into the statistical part of the thing.
Okay.
Sorry, I think it's required because you did an ANOVA.
Yeah.
Which are the factors?
The two-way ANOVA.
Two-way ANOVA.
Depth and distance, yeah.
Different distance, and which are the levels of those?
So it kind of depends on what you're looking at, but the depth would be in 4 categories of 20 meters. Yeah.
Why?
I have to be honest, I did the statistics in February, so it is a bit in the back of my brain.
Yeah.
Yeah.
I think you're trying to categorize two variables that are continuous.
Yeah. I did not have.
To be a problem.
Yeah.
It's better to take them as what they are,
Yeah.
continuous variables, and if I don't know, you will do a regression or BLMs or something like that.
Yeah.
Because they are, you need to give
But I did do a general linear mix.
You did?
Yeah. Yeah. That's the graph with the lines. Those are regression lines.
well, why did you switch with ANOVA?
I think in the end, to be honest, think in the end I didn't do an ANOVA, I did the general mixed
Ah,
mode.
okay, that's
Sorry,
confusing.
yeah, that is confusing. I think
Yeah.
I just put it wrong in the slides then.
Yes, and can you go through those graphs of the effects on biodiversity, like depending on distance to the glacier and
This one?
these ones? I would have made these ones, this graph, bigger so we can see it better,
Okay.
and put the dots, the points.
Yeah.
And this is important to see by the width of the confidence interval. I think you have very good relations.
Yeah, they're very
But put the dots so you can see it.
okay. Yeah.
So can you explain this again? Because you have an increase in biodiversity very close to the glacier,
In the shallow waters,
in
yeah.
the shallow water.
There was more species richness than in shallow waters,
For
far
benthic-
from the station.
For benthic community,
Yes,
right?
yeah. That was very unexpected.
Mm, maybe you should have thought in spatial analysis, because a given point in a space can present some characteristics, not because the variables change there, because there is another point in a space that present the same. This is called spatial autocorrelation.
Okay.
So one point can have some characteristics just because it's close to another point.
Okay.
And you need to correct for that. You know what I mean?
Yeah.
And these effects could happen.
All right. That's very good feedback. Thank you.
Yeah, I think that's all from my side. Thank you very much.
Yeah.
And congratulations.
Thanks.
Thank you, Nico. I'm sure Mother Nico has lots to say.
Yeah, always has.
Yeah, I guess I'm gonna keep torturing you a little bit with the stats.
Bring it.
And then,
It's fine.
But I think you mentioned in your introduction that you did the CCA as well.
I did.
Okay, and
I know,
it's not
because
there.
of time I couldn't put it in, but
Okay.
yeah.
So probably, I mean,
What if-
make the presentation self-consistent.
Yeah.
Because I love materializing this now.
I can't tell you what we found, but we found that close to the glacier was not a very clear pattern in the mobility of species, but the further away you got, there were more mobile species. And then there were
Also,
three
specific
clusters.
parameters traits. Okay.
Yeah, and there are three clusters of the stations, two different close to the glacier clusters, and then one with all the rest of the stations. Yeah.
Interesting. I had a little, it's more of a little question. You mentioned that you had some abundant diatoms in some areas.
Mhm.
And you mentioned that they might be indicative of change, some change, if I remember well.
Yeah, with diatoms.
And I'm wondering, oh yeah, you actually
Yeah.
see it. And I was wondering, why diatoms are specifically indicator of system change. In that context, I don't know necessarily the system, so I don't necessarily see. For me, I'm a quantum ecologist, so I'm just putting on one.
Yeah.
So how are they an indicator of system change?
Well, there's some recent papers on the diatoms that if they bloom in an area, then later they find a different ecosystem. Probably because the primary production just gives other species a bigger chance, and it makes a bit of shifts within the ecosystem. That's what they found in the Western Peninsula. I don't know about the rest of the world. Yeah.
But yours was one time point, right? It was one campaign at one time you went there.
Yeah.
So I was wondering, maybe they're just normally blooming in this ecosystem every year.
Yeah.
So that's what yeah. Okay. I have a more general one, so I don't know if you have
You have plenty of time.
but yeah, so you have this pattern that seems pretty strong of more alpha diversity generally near the glacier, which you were not expecting. Which is understandable. I mean, I really like the way you introduced your hypothesis at the start. It was really nice to have that. And so I wonder if you are familiar, if you heard the intermediate disturbance hypothesis. which is quite a classic thesis in community ecology, but it's more used in terrestrial ecosystem often, so in marine we have
Yeah, there's a really great study by the Koreans on Marion Cove where they could basically form 4 clusters in the same area, and then highly disturbed, intermediate disturbed, and then deeper depths.
Yep.
So yeah, I've heard of that, but I couldn't find a pattern as clear as they did. I think it's because Collins Glacier has been more compact. It has receded a bit, but it's not as much, whereas Marion Cove, half of the cove has disappeared in glacier size.
Okay.
So their succession is very different. Yeah.
But do you think it still could be explained by some disturbance by the glacier, but it's like not high enough to bring your alpha velocity down, and it's actually bringing it up because it helps to avoid yeah, because you mentioned succession, so do you have again, I don't know this system, but do you have what you think are later successional species in deeper area further away from the glacier?
Yeah, there's a bit more sponges there. Yeah.
So actually, yeah, that could work with could be consistent with that.
Yeah. Okay.
You good? Yeah. Well, we still have time. I might also have some questions.
Okay.
Thank you for your presentation. I'm also very interested in the food webs in the Antarctic Peninsula, so I was really interested in that. Maybe I'm mistaken, but I have the feeling that you did not really explore beta diversity beyond functional diversity.
No,
Did
that's
you not
right.
try to compare the community composition instead of biodiversity and disease?
No, that's not what I did for this. Mostly because I was focused on doing a baseline study, so
Okay.
I just focused on the alpha biodiversity a lot. Yeah.
Okay. And I have some questions about your trophic groups, which is mostly what I'm interested in. Can you tell me what the difference is between filter feeder and suspension feeders?
Yeah, filter feeder is more connected to the sediment, whereas some of the cities that we found were almost a meter up in the water column.
Okay.
Yeah.
So it's not really related to how they feed, it's related to their environmental position. And opportunists?
Would mean that they could scavenge, but they would also do any kind of feeding that is possible. Yeah.
Okay, well that's good for me. Can maybe the room has some questions?
I'd like to presentation. Thank you so much. I was just wondering, you mentioned that chlorophyll was higher and you related it with the abundance.
No, I don't know if chlorophyll was higher. We have hypothesized. Sorry? Yeah.
Yeah.
We hypothesized that it is, but we can't be sure because we didn't do water
So
sampling.
you didn't measure chlorophyll?
I don't know. No.
Because in general, it wasn't case of glacier, and like general coastal area is higher because of the other thing, but in glacier it's lower. And you said that it's higher, and maybe that's the reason for the abundance, higher than the relative abundance.
Yes, that's what we think too. Yeah. Thank you.
Any other question? Yeah.
I was wondering what the hardest part of the sampling was.
The weather. I made something for this. Good question. Let's skip through some videos. So in November, I had to make a planning of my sampling. I would arrive December 3rd and leave December 20th. I would do, if I was lucky, 2 samples a day. And then I had some days where I would not be able to sample because of the weather. We also couldn't sample on Sundays. So this was a very nice plan. I was very ready to do this. And then this is what it ended up being. The ROV broke down 4 times, the boat broke down, there was storms, so we had to sample on Sundays, which they were not very happy with. There was a lot of mist, there was of course now Christmas and New Year's involved, so there were 3 days that we couldn't work. And in the end I stayed until January 4th, and it took a lot of extra work, yeah.
Thank you so much for your presentation. It was really interesting. I'm just wondering what you think might happen in the functional ecology in future climate scenarios with the ice caps melting. and how fast those changes might occur?
Well, that's what I'm going to do my PhD in, so I'll know in a few years. Yeah.
Yeah, really.
And maybe I missed it, but maybe you could say like what the most important species were and why do you think, like what does it say about the ecosystem?
So it's the Nemertea. It's probably because they're able to scavenge and also hunt. Plus, they're quite mobile. They're found all over Antarctica, basically. Yeah.
Okay, right, another one?
Yes. In the winter time, there will be no melting, or hardly any melting, of course, and then the amount of salt in the water will increase in Collins Bay. Maybe it will be frozen over. How do you think will affect that? The biological communities.
Well, most of these benthic species are seasonal species, so they're not going anywhere. The production might be lower or changed, but that's what I would expect.
So no effect.
No, not on the. Okay,
Thank you.
thank you.
Just wait a bit so that the online audience.
We're a few minutes ahead on the planning, so we have to wait so that people online don't arrive when the presentation is already started. You have to right? Okay. When you only have 2 minutes left, I will wait with you.
Yeah. Perfect, thank you.
Try to attract your attention so that you know that
Yeah,
it's
no,
time
perfect.
to start concluding your presentation.
Yeah, that's good, thank you.
Right, guys, we'll start in a few seconds, please. Okay.
whoever wants.
Good morning, my name is Erin Villanueva Coronado, and I'm going to present my thesis about assessing temporal and seasonal changes in epifaunal communities associated to Solstera marina in the Skagerrak region. This research was conducted in the Institute of Marine Research and supervised by Corinne Gagnon. Seagrass are marine flowering plants that grow in shallow coastal waters. In Norway, the dominant species is Solstera marina, also known as eelgrass. These seagrass meadows are important due to the ecosystem services. As they improve water quality, they act as a carbon sink and carbon sequestration. They stabilize sediments and act as a coastline protection. They serve as nursery areas for juvenile fish and important commercial species. And they serve both as food and habitat for a diversity of organisms. However, it is continuously threatened by overfishing, eutrophication, habitat fragmentation, causing a worldwide decline in seagrass meadows. The organisms living in the seagrass meadows can be divided as infauna, the ones that are below the sediment, and epifauna, the ones that live in the seagrass plates and above the sediment surface. These organisms play an important role as they graze on epiphytes, like for example the mesograzers, which maintain the seagrass health. They support food webs, they help in the recycling of nutrients, and as they are sensitive to environmental changes, they are a good indicator of ecosystem health. In the Skagerrak region, which is where this study will focus, it has been documented some epifaunal community changes. In Norway, there was a pilot study, and also in Sweden, where it has also been documented a decline in key mesograzers, like for example, amphipods and isopods. Most of the studies, however, has been focused on summer, leaving out all the seasonal changes, which is what this thesis aims to address. The objective of this thesis was to assess the temporal changes in the composition and diversity of epifaunal communities associated with Solstera marina. This thesis builds on historical data of 1999 published in a paper in 2005 by Frederick Schwann et al. And it focused on 4 research questions. Whether or not the epifaunal composition, density, and diversity have changed from 1999 to 2024, how the seasonal patterns differ between years and sites, what drivers could explain these changes, and what are the ecological implications of these changes. This is part of the Norse project, which focused on seagrass in the Skagerrak, Kattegat, and Baltic Sea. The study area was in southern Norway, which is part of the Skagerrak region, and it consisted of 2 sampling sites, Flava, which was in a sheltered area and also close to a river outlet, and Europa, which was in a more exposed area. We also had access to monthly data of environmental variables in the arability station, which was in the middle or in the medium point of Flava and Europa. For the sampling collection, we based the methodology in the previous paper published, as we aim to replicate this publication. So the collection was done in 4 months, April, June, September, and November. It was done by scuba-diving, and I will explain the main differences between these 2 sampling collections. So in 1999, they took 3 replicates, and what they did was taken a quadrant, pregnant the surface, and collect all the eelgrass in that quadrant, and put it in a mesh bag. In 2024, what we did was taking 5 replicates instead of 3, and we collocated the mesh bag on top of the seagrass, of the eelgrass, and collect all the eelgrass and the epifauna associated from the cutting the eelgrass from the rhizome. After collection, we took the samples for the laboratory analysis, where we rinsed the samples with fresh water 3 times, then we sieved it with 2 different mesh sieves of 250 and 500 micrometers. We preserve it in ethanol, 70%, stain it with rose bengal to facilitate the sorting, and then under the stereo microscope, sort it and identify it to the lowest taxonomic possible level. For the data analysis, first we needed to standardize the area, since we have different sampling area in both samplings. And for that, we calculated density in milliliters per meter square. We also calculated diversity indices. And to test for differences, we used a GLM and then did a post-hoc pairwise comparison. Also, we separated the organisms in functional groups based on feeding modes: grazers, predators, filter feeders, and deposit feeders. We did an NMBS and a Terminova. For the long-term changes in epifauna, in this graph we can see the mean density, the kilos per meter squared, but by month and separated into two sampling sites. And in blue we see the data, the previous data, and in orange we see the data from 2024. So first we see in Java, for example, that previously it had higher density, especially in June where we see a peak, and it has decreased for 2024. In the case of Landurumba, we see the opposite. It used to be low density, but now we see an increase in the total epifauna density. When we tested for differences, we found a significant three-way interaction between years, month, and site in the total epifauna density. We also calculated the diversity indices, and the one that we saw clear patterns was in species richness, where we saw a reduction. And we also found a three-way interaction in species richness. To compare the community composition, here we have the relative density in percentage of the major taxa, like for example Ficora, bivalves, and others, and it is also separated by month, the 2 sites, and the 2 years. So in 1999, we see that it was dominated by bivalves, shown in this orange color, and then for 2024, we see a big decrease in bivalves, specifically Mytilus segulis. In the contrary, we see an increase in Gastrocoma, for example, in the light blue, and in the case of the Europa, protocols, and nematodes. For the functional groups, we also saw that the suspension figures decreased from 1999 to 2024 as expected because of the decrease in Mytilus segulis, and we see an increase in the green color, which is the grazers and the deposit feeders. We also performed a non-metric multidimensional scaling, and even though there are many overlaps here, we do see a difference in the years. So here's all the data from 2024, and here's the data of 2099. When we did the Terminova, we found also a three-way interaction between year, month, and site. So, regarding seasonal variability, which is one of the aspects of this thesis, we expect the seasonal variation as it affects both the Zostera marina biomass, the habitat itself, and the epifauna that lives in this habitat. And we saw this variation in the total density as shown in the previous graph. So we saw a peak in Cava in June, and in Landurumba we saw a peak in September. These were, even though I didn't show the graph of biomass of Zostera marina, these were also linked to this. So where we saw an increase in June, there was also an increase in biomass and total epifauna density. And this can be linked to the habitat complexity of Zostera marina, which affects the food availability, the shelter, and colonization. And all of these factors are important to study as a multi-seasonal sampling to capture the full variability in the year. What could be the drivers causing these changes? So in Sweden, there has been a study published where they explain the top-down control. Due to overfishing, the top predators have decreased, causing an increase in the intermediate fish predators, which also caused a decrease in the next level. So the mesograzers have decreased, the small epifauna, and it has increased in the large epifauna. Another possibility is bottom-up control. So in previous studies, it has also been recorded that the nutrient enrichment and early stages of eutrophication causes an increase in filamentous algae, and this also causes an increase in epifaunal evidence. It's important to know that this is at a certain point, as if it continues to increase, it will cause anoxia and a decrease in the organisms living there. What we saw in this study was an increase in grazers and deposit feeders. In terms of environmental data, we saw an increase in chlorophyll in April and September, a decrease in water clarity and oxygen. So we linked it more to our nutrient enrichment situation rather than a top-down control. What could be the ecological implications of this? So, nutrient enrichment can cause eutrophication, which affects the light as it increases the filamentous algae, reducing the light for Sostra marina and affected its health. It can cause faunal shifts, which will change the traffic transfer and food web. And in that context of climate change and global warming, it is important to know that both of these threats can cause a greater decline in seagrass health. For future directions, it's important to test whether eutrophication is causing these changes in epifaunal communities. And since this is site-specific, it's important to do it also in southern Norway, because there are other studies, but not exactly in the site of study. For the conclusion, epifaunal communities have changed significantly in the 25 years. The seasonal patterns are site-specific and dynamic. We also see functional shifts suggesting early stages of eutrophication. Important limitations to note is the sampling area, which was different between the two sampling collections. This was addressed by calculating the density, but still it's something to note. We also didn't have environmental data of these sampling points, as it is in a monitoring station in the middle of the two sampling points. So we don't have exactly what is happening where we collected the Sostra marina, but it shows some of the trends that can illustrate what has happened in the last 35 years. For the value of this study, it contributes to the NORSE project, and it contributes with long-term and seasonal data for this. It's important to note that multi-seasonal monitoring captures site-specific dynamics, which are often missed in summer surveys. Thank you so much for your attention, and I want to thank everyone in the Institute of Marine Research, my family and friends. Thank you.
Thank you very much. Who wants to start? No?
Thank you. So thank you very much for the presentation. I really enjoyed it. Probably I lost something during your presentation, but I'm wondering, did you find also different kind of species in the 2 years? I mean, the fauna composition was almost the same, or did you find also new species present, or maybe invasive species that in the previous paper they didn't found?
We did see different species, as I showed briefly in the major taxon graph, but we found 24 new species for this study, so which were not found in the previous one. And another thing to note is, even though we didn't find the only we saw both species that were not present anymore, and we saw an increase, for example, in amphipods, which were not that many in 1999. So there were changes, there are shifts in the epifaunal communities in these 2 years.
Did you see if, for example, some of these new species that you find are more linked to warmer water? I mean, that moved from the south and...
I did not look specifically into whether or not we're from warmer waters, but as far as the species list we had, it was from the area still, just different species from what it was found before.
But more or less native species?
More or less, yeah. Yeah, not...
no alien species or species that are more linked to warm...
Not that I remember, and as far as I looked for it, not.
I also wonder if you found strong degrees in the Meteorus
yes.
Specimen, the Meteorus, and I think that well, it's also happened here, probably you know, and we think that is linked to the increase of temperature, to the global warming. Do you think that could be also linked, also they are, to be due to the increase of temperature in the ocean?
It could be a big possibility since it's happening also there. However, when we compare the temperatures, as I mentioned before, the environmental data that we got was from a monitoring station, not exactly in the sampling point, so it doesn't reflect exactly what is happening there. But still, we didn't find any big difference between the temperature in 1999 and 2024. However, it doesn't reflect exactly what is happening in the sampling sites. But it is a big possibility, and for previous studies, it is still something that is in debate on what is happening exactly here. It has also been recorded in Sweden that there was a big decline. In the pilot study was also recorded that there was a big decline in methylocedulis. So this is what we see here. That, yeah, there is.
Thank you very much.
Thank you. Nico? Sure. Well, Aaron, congratulations. I really like your presentation. Perfect combination in between figures, very nice maps, pictures, I really love it. Very nice. Congratulations for that. Also very clear explanation on everything. You're present everywhere, listening to the public all the time, pointing the relevant points there. I mean, very nice, really. Also, the statistical design, statistical procedure seems pretty clear and solid. Congratulations for that too. I just want to ask you about interpretation of the result, because here you are just getting two snapshots. You cannot look at trends with two snapshots. So all the climate change theoretical impact on your results is just an hypothesis.
Yes.
No? But I wonder, this is part of a bigger project, so I guess in that monitoring station they are sampling, I don't know, every month, every year, or so. Do you know that?
For the NORS project, how frequent it has been sampling?
A particular monitoring place.
It has been sampled 2 years ago in a pilot study. It only included June and September, and we saw similar results for that too. And before that, as far as I know, there's not other monitoring in those specific places because it has started quite.
But is there any specific monitoring place where they have sampled recurrently from the 1990s to now?
In southern Norway, as far as I know, not exactly, not in this area at least.
Yeah, because it would be nice to
compare
your results with that, because this points to climate change, but it's not clear. There are interannual variations that may not be, at least they're directly linked to climate change. I mean, you're in a very dynamic region from the point of view of circulation. Some years you can have more entrance of water from the North Sea, or the opposite, more influence of the Baltic.
Yeah.
Do you think your results can be explained in that way?
First, just to point out that NORS project aims to address this, so they are now starting to get more data and contribute to all these big picture projects. So it is an ongoing study and an ongoing project that will later be published and later continue to be analyzed. And in terms of the variable or the variability that can be explained, as you said, it is an area that can be influenced both from the North Sea and the Baltic. But these sampling points, as I mentioned, one was like in a shelter area, one was in a more exposed area. So even within this small scale difference, spatial difference, we see different results as in this image, and
yeah, but also it could be a change of substratum.
Yes.
I mean, that is what the presence of muscles indicate, because muscles mean something like that. Probably there was some rocky substrata there, right, in the past, and then probably was covered later on by a thicker layer of sediment. So you will have other species more linked with the same. Could that be the something? Could that be?
Could be an explanation.
Because the muscles have to be attached
Yeah.
to something. They don't grow just in pure mud or sand.
Mm-hmm.
And the changes in sediment can happen very fast.
Yeah.
From one
Those are
season to
changes
other.
to, yes. Those are changes to.
It's just, climate change is behind many things, and probably it is behind this too. But I would be, I would have caution to address,
Yeah.
to link both things.
Yeah, no, and also for this, to answer more of these questions, even though it's not the same to do an experimental study, it can be studied that way too, like a field cage and controlling some of the factors to see if this is what is changing. And it's important to know that this is site-specific as well. So even though there are studies in Sweden, for example, it should be done also in the area that we're sampling now, because it is different conditions, different species that are previously living here that can be affected in many ways.
Okay. Thank you very much, Erin. Congratulations.
Nico. Thank you. I was wondering, still coming back a little bit actually, and the conversion of the two years. So you have two slightly different sampling methodology. How do you think that could affect possibly the results?
One thing to note, first sampling area was different, we tried to address it, but here for the Ecuador it was 0.25 meters squared, and for the new sampling was 0.071 meters squared. So the sampling area was different. That is one point. Another point is that methodology itself was different. So here they used quadrants, and then all of this was with the mesh bag and cutting from the rhizome. But this can cause a lot of use of some mobile organisms, which was tried to address in this one, but the mesh bag was put on top of the surface, cut the rhizome, and try to capture as much as possible. So there some of the changes in the epiphytal communities can be also linked to this, because it was a different methodology. There was a different sampling area, and it was a different methodology, and it can affect, as I said, the model fauna found.
Yeah. Okay, yeah, makes me feel more fauna. And you mentioned the different surface areas. That's a general thing for when you do diversity studies. What do you have an idea of an important aspects that shifting your sampling area could change in your results? An important aspect of community that we often look at, and that is very, very quickly and very heavily impacted by the area you have.
Yes.
I have one specific thing in mind that you might have.
No, it is something that we consider. We try to do the species accumulation curves to see if the sampling was enough or not. But yeah, it was something to have considered, and it's one of the limitations of this study, that we use a different sampling area.
Yeah, you mentioned the species accumulation curve. That's exactly what I was a bit worried about, the self-diversity between two species. The more you sample, the more you see rare species.
Yeah.
So, yeah. Okay. I also have a bit of a technical question on one of the statistical criteria you had. Your LNDs. On what distance is it based?
This was based on Bray Curtis. It was based on Bray Curtis, yes.
Were they computed from the raw balances or from the relative balances?
So it was computed from the raw abundance. Something to mention here is that the reason why we see only 1 point for 1999 is because we didn't have access to the raw data of the previous study. We only had access to the published data. That is another limitation of this study. That for the 2024 data, it was of all the replicates, the raw data with all the replicates. So here we can see for the same site, the same month.
Oh yeah, so you have no
yes, here we don't have the replicates, here we have the 5 replicates. That is also another difference, yes.
Yeah, I think generally when you do any kind of NDS or something, application, you've got to state from which kind of distance it's computed, because that's super important for application. I don't know.
You can try asking questions.
Sorry. So another one I had was on the environmental factors.
Yes.
So you tell that story about the potential effect of certification, but I haven't seen any like more formal tests and graphs and relationships to explore. I mean, to explore those relationships. Is there a reason for that?
It was mainly due to time that I didn't show the environmental data. I just mentioned the most relevant aspects for this study, but we had of temperature, as I mentioned, it didn't show a difference between the years. Nutrients, also there were fluctuations, but it was not a big difference between years. So that is, I didn't, as I said, I didn't show it. Chlorophyll, we did find 2 clear peaks and more increase, and oxygen decrease, second water clarity decrease. So yeah, regarding linked it to eutrophication, and in the nutrients we saw fluctuations. So it has been an area that has been greatly affected by eutrophication, but it has taken some measures to prevent it.
Last one? Yeah, I guess in terms of specific, coming back to the eutrophication issue and another question. So generally you have more abundances in 2024. The abundances of the whole communities.
yeah,
They are more abundant in 2024, generally, no?
it is less for clava. We saw a decrease in clava. So generally we saw a decrease.
Oh yes, that's different on site. Okay.
Yes, it was site specific. So in clava was decrease, in Europa increase, as we can see here. And as I mentioned before, even though the environmental data is not exactly the sampling point, that doesn't reflect exactly what is happening here. So in Clava, it is in a shelter area close to a river outlet, so the environmental data will be different than in Lanjarumpa.
Yeah, because I was wondering, especially for those ones, so you normalize, you made a density, so that's as a function of the ground area.
Of the sampled area?
Yes.
Yes.
And I was wondering if you see a possible other way to get density that would be very relevant for the community, and that you could have possibly included, I mean, if you have the data, especially in the line, but another kind of normalization to get density that would be very, very relevant.
Apart from density calculations, I'm not sure.
It's another density.
Sorry?
Where do they live?
Sorry?
Where do they live? Okay.
Yeah, we didn't do it based on this. Yeah, that could have been another way of doing it based on that.
Because that's, in a way, it's very much linked to your environmental variables, because you could have changed in the very habitats that they live in, and yeah, that'd be better.
Yes, even though we didn't do it by leaves. Okay, no, it's okay. I was going to say that we need to take into account biomass of Zostera marina. It is not exactly leaves, but we did see a link in the increase of Zostera marina biomass. For example, in Clava, it was in June, and we also saw an increase in the epifauna. In Lanjarumpa, there was an increase in September, both in epifauna and the biomass of Zostera marina.
Thank you. There's time for one small question from the public, if you want. I think they all want to have their break.
Thank you very much for your program.
Good. We're live. Yeah.
Okay, so welcome to the final session. 15 minutes as usual for presentations, 15 minutes for questions and answers. In the jury we have Lars, Gauthier and Esther, so thank you for joining as well. We have Madalena up first to present your thesis research, so over to you and the very best of luck. Thank you.
Hi, I'm Maddie. I'm going to present my thesis on quantifying the behavior of Atlantic bluefin tuna during rod and line capture that I did in Bergen. So I'm going to introduce this to you from the lens of welfare, which is what got me interested in this topic. As I will talk about, wild capture fisheries represent a big gap for welfare regulations in the food that we consume as humans. So before I move on, I will just talk about what welfare is. So there are these 5 freedoms that were established in the 70s for terrestrial livestock. Basically that every animal should be allotted these 5 freedoms at all times. There's a complementary 3 general approaches to assessing welfare in animals. So the nature-based approach talks about how animals should be allowed to live a natural life and express natural behaviors. The feelings-based approach says that welfare depends on how an animal feels, and the function approach emphasizes more the biological system that it should be functioning properly at all times for good welfare. So as I briefly mentioned before, there is a welfare gap in this major global food source. This is the most recent FAO report. We can see that capture fisheries and aquaculture are representing roughly the same amount of global production of aquatic animals. So in aquaculture, as well as in terrestrial agriculture, there are welfare regulations in place to kind of safeguard these animals that we consume, but there's nothing at all for capture fisheries. So it's about 1.1 to 2.2 trillion individual fish that have no welfare regulations. So it brings me to catch welfare, which is a framework that talks about why and how we should implement welfare into fisheries. It has been studied in other gear types and other species that are different from what I did in my study. So this is specifically mackerel and purse seines. Purse seines, if anyone doesn't know, is a gear that catches many pelagic fish in a big net in a large group. So the main stressor there is crowding. So we know that acute stress pre-slaughter does reduce the flesh quality. You can see in the top image some gaping in the flesh. That is a direct result of capture stress, and it will reduce the price of the fillet at the auction. Compared to the bottom photo, we see a low stress situation. There are 3 main methods when it comes to assessing welfare in capture fisheries that have to do with the stress responses. So the primary stress response is neuroendocrine and has to do with an animal's fight, flight, or cope response to a stressful situation. The secondary stress response are physiological, so the metabolic changes that occur inside the body in response. And then what I focus on is the tertiary response, which is behavioral. So it's an animal's overall reaction that is observable, non-invasive. Some background on bluefin tuna. So they recently in 2021 were moved from endangered to least concern on the IUCN Red List. This is the global stock. So the tuna fisheries are in recent years ramping up once again, bluefin tuna. It is an incredibly economically valuable industry. A single bluefin tuna can be worth millions of dollars at the auction. And for all fish, prices dependent on quality, but especially for bluefin tuna, it's really important that they're a high quality. Just a brief background on bluefin in Norway, as this is where I was focusing. The east coast of Norway is their northeasternmost range, and they go there every fall for feeding. They did a lot of purse seining up through the 70s, and then the tuna numbers kind of declined there. So the fishery was closed for a number of years and then reopened in 2014, still the purse seine fishery. And then more recently, the scientific tagging programs have been implementing rod and line gear. They've had a lot of success with that. so it has actually started to establish a commercial routing line fishery that had 22 vessels in 2023. So routing line is a gear that inherently catches fish one at a time, which is very different than other gear types. This method allows you to individually assess every tuna that comes on board for welfare, so it really is well positioned to adopt welfare conscious practices, maintain high quality, and get a higher price. So my research objectives, I wanted to characterize the behavioral response of bluefin tuna to routing line capture, as well as try to quantitatively describe that response through relationships between the behavior that I observed and the data I got from my accelerometer. So how did I approach it? The project that I'm working in is called Well Tuna. The Institute of Marine Research also has a tuna tagging program that's part of the International Commission for the Conservation of Atlantic Tuna, ICAT. So their tagging program, we kind of collaborated with them and we were able to attach my cameras and accelerometers to their gear. And in September 2024, they caught 6 fish from which I used that data. The way they fish for these tuna is on small coastal power boats. They troll this gear through the water on these spreader bars here, which look like this up close. So I attach the camera here, looks down over the gear, and then the tuna would be attached on the hook on the other end, and with the accelerometer attached very close to the hook to try to get a reading of the tuna's movement. So on to data processing. For the accelerometer, it records acceleration in 3 planes, x, y, and z. You can combine the data on those 3 planes into one value called overall dynamic body acceleration, which is commonly used to estimate energy expenditure and has been done in multiple terrestrial species as well as other fish species. However, normally for that, the accelerometer is attached to the body of the animal. Because in my case it wasn't, I approach it a bit more conservatively and I talk about it in terms of activity levels instead of directly energy expenditure. For my video analysis, as I said, I had 6 capture events, so 6 tuna. Each one ranged of a fight time between 10 and 20 minutes, so that's my full video duration for each fish. From that, I cut the whole capture into 5-second clips, and then I analyze each 5 seconds for a behavioral quantification. I had 7 metrics, 6 qualitative, 1 quantitative. I will give a bit more information. So for my statistical methods, I wanted to see if those behavioral observations that I showed in the last slide could predict the overall dynamic body acceleration to see how well behavior is correlating with the physiological response. So I created a generalized linear mixed model. Those behavioral metrics were the predictors. The response was the ODBA with a random effect of the capture event because I'm not interested in the differences between the capture events. I then included all of them as well as interactions from the beginning, and I did backwards selection and only retained the significant predictors. I also had to add this Ornstein-Uhlenbeck process just to adjust for temporal autocorrelation, which is necessary for the behavioral data that I had. All right, so on to some results. I'm going to show you what some of my videos look like to give you an idea of what I was working with. So this clip would have been the very first clip at the start of capture, right after the tuna bites the hook. You can see there's a lot going on. There's a lot of bubbles. It's moving very fast. You can't really even see the fish. In the bottom, also, we have quite a fast, intense movement period. He's going deep. I can't see much. This shows an example of the tuna going back to the school, which is not something we knew that they did, so it's actually quite interesting. And then this video on the far right shows just an example of how I was able to get tail beat frequency out of these videos pretty clearly. So the general trend that I saw was a very high activity level immediately post-hooking, and then a very kind of quick, relatively quick decline towards a stabilization of a tail beat frequency around 1 Hertz, with some intermittent peaks throughout the remainder of capture. I had a tail beat frequency maximum between 4 and 6 Hertz across the 6 fish. 4 out of 6 of them also went back to swimming with the school, like in this picture, and both of these things always happen right at the start of capture. So here I'm going to present my kind of ethographic behavioral results. It looks more complicated than it is. So this is just showing other tuna, if other tuna are present or not. On the x-axis we have time bid, so it's every 5 seconds, just for one fish. And then I recorded if they were changing direction or not. The orientation in the water column, how they were interacting with the gear, a rough tail beat amplitude, tail movement style, and lastly tail beat frequency, which was my only quantitative measure. Where you see gaps here, it's because I wasn't able to see anything on the video. So this is just for one fish. This fish had particularly good spread of videos that I could observe, but not all of them have this much data. All right, so for the results of my model, as I said, I included everything. I did backward selection and only kept the significant predictors, which were direction change, tail movement style, and tail beat frequency. Based on likelihood ratio tests, you can see that tail beat frequency is really the main driver of ODBA. It's accounting for the majority of the variation in that response variable. This is a visualization of the model. So the main result here is that my observed behaviors can reliably predict ODBA. We see a positive nonlinear relationship between tail beat frequency and ODBA. When it changes directions, you're seeing it's predicting higher ODBA. What's not immediately clear is that intermittent tail movement also was predicting slightly higher values than constant. The model fits a bit better at lower tail beat frequencies than higher tail beat frequencies. So yeah, back to the behavioral profile. This high activity that I observed at the start is consistent with an observation by Wardle in 1989, where he observed a maximum tail beat frequency of 6 Hertz while he was feeding bluefin tuna in sea cages. So that's consistent with what I saw at the beginning of my capture. He also estimated a theoretical maximum of 10 Hertz based on muscle contraction rates. The remainder of capture, I have this stabilized activity around 1 Hertz, which is consistent with routine swimming values that they've gotten from post-release studies. And then at the end of capture, it's something I see on the video. It's also something the fishermen have observed, that by the time the bluefin tuna are coming back on board, they're really not fighting, they're not really resisting. It's very low activity levels at that point. So briefly to the question, is capture stressful at all? How stressful is it? Or are they maybe more relatively unaware that they have even been caught? So I think what my results are showing is more of a normal, natural predatory behavior that's triggered by the prey encounter, as opposed to a stress response to the capture process. I think this for a few reasons. This elevated initial activity is consistent with what Wardle observed during feeding, so we know that they have this fast, high activity reaction while feeding. Also, this really didn't last long at all, and probably was never more than a minute in any of my fish. This decline to stabilization, the video shows you that's routine swimming, that's 1 Hertz. So we know that they're exhibiting normal swimming behavior most of the time. When they return to the school, I think it shows that they're actually returning to the behavior they were doing before they were even caught. And then lastly, they have really exceptional metabolisms. So they have a high cardiac output, they exchange oxygen really efficiently. It would take a much longer period of high activity to truly exhaust them. So some practical applications of this work. If we can understand the behavioral response, we can build a baseline for good welfare. In theory, fishing techniques that could minimize tail beat frequency, direction changes, and this intermittent tail movement that I saw from the model would get lower ODBA, therefore a lower physiological internal response, and improved welfare. So the main takeaways here are that my behavioral metrics shows that behavior can predict ODBA. I think that it's more a natural sequence of predation rather than a response to capture stress. That's telling us that rod and line gear might have really strong implications for maintaining good welfare. Right now, this is not the most common type of gear used for bluefin tuna, but I think it has a lot of potential. So next steps. If we can get a lot more data into this model and make the relationship between behavior and ODBA much stronger, the idea is that maybe in the future we can kind of reverse the model and be able to predict behavior just based on ODBA. And then we can establish some kind of thresholds here for maybe in real time you can see, oh, this tuna is is at a point of poor welfare, and you can make decisions based on that. I was also the only one that observed my videos. With more observers, we could assess stronger reliability of the method. I also thought it would be nice to expand into different fishery sectors and see how commercial fisheries and recreational fisheries, the gear and the methods that they use, how is that affecting or changing the stress response? And then lastly, of course, an obvious next step is to take some physiological measurements, do some blood sampling, postmortems to kind of try and ground truth the behavioral response that I'm seeing. And that's all. So thank you to my supervisors.
Well done, Mary, thank you. So to our jury members first, we have questions. Whoever wants to start.
Yeah, sure.
Gossip.
Always have questions. I don't have many questions because your presentation was really super clear. So I'd like to congratulate you on that. I wanted to talk about this behavior of returning to school. It was not clear to me how you interpret that.
So, in this type of fishery, actually, that I was working with the scientific tagging team that does this fishing every year. They had no idea the tuna were going back to swimming with the school, which I clearly see in my videos, obviously, as you saw, right? So I interpret it more as, I mean, there are a couple of different interpretations. It could be that they're kind of seeking group safety, so maybe they do know something is wrong and they're going back to the school for that reason. I think, though, it's more because they go to Norway for hunting and feeding, which they do in schools. Otherwise, bluefin tuna are not always a schooling fish, but when they're feeding, they are. So I think it's more, you know, they were swimming with the school before they got caught. They tried to encounter a prey, and then they go back to swimming with the school.
They realize there's something stuck in their mouth.
I don't think they do. They could, obviously, I don't know. What can I say?
You see the spikes and here and there, it suggests that
yes.
they are reacting.
It suggests, yeah, it does suggest that maybe there's an intermittent escape response, kind of. The other thing is, I only had 6 fish. I don't see this consistently across every fish, so it's harder. I feel that it's harder to draw that conclusion than to actually say, well, maybe they don't even know. Maybe that this is just normal behavior.
What would you consider the behavior that would be indicating of stress?
I think if we saw a longer period of elevated activity, that would be more indicative of an escape response where clearly they know something's wrong. So they're increasing this activity level, which I would see in the tailbeat frequency. I would also see in the acceleration data. I know I didn't talk too much about those numbers actually, but ODBA was fairly low, I believe.
Have you ever seen videos of looking tunas in bursting?
Yeah, I have.
It's super quiet.
Yeah.
And until there's no more water between tunas, it's very impressive. It's super quiet. There's no splashes in the sea.
They're a pretty impressive fish. Yeah.
They're hard to stress animals.
Yeah, I think so too. Yeah.
Or they hide the stress very well. Thank you very much.
Thank you.
Thanks.
Yes, for the team, the super team, I have a question about maybe basically, what could you do? Is it possible to add some acoustic survey to because when you say they return to the school, the range that the camera can see underwater is rather small. So how would the acoustic be useful in such study?
I don't know that much about acoustics, but I think it would be useful to track. I think it would be interesting to see how far an individual tuna actually gets away from the school when they attack the gear and get hooked, right? Because it seems like on the videos that they're never really that far away from the school. They always, after they get hooked, they are going very quickly back to the school actually. So I think it would be interesting mostly to see the spatial dynamics of the other fish.
If the boat is going to another direction than the school, the fish is not trying to resist and go back to the school.
So the method that the boat uses is not fighting against the tuna. I didn't have time to talk about this, but these fishermen, they let the tuna run when they catch it. Because really there's not a lot of point in fighting a bluefin. You're probably not going to win. You're going to exhaust the fish more. So what they actually do is they let it run, and they position the boat in a way that it can easily run out this line until it gets to they take their moments to reel the line back in. So they're never trying to really fight it. But this is also the protocol of the scientific fishing. So that's why I think it would be interesting to see it in other sectors where potentially they are fighting the fish more. So how does that affect the stress?
Yeah, okay. And then, what other possibility do fishermen have to catch this blue tuna fish?
Like what other gear?
Yeah.
What do you mean? So it's more common in purse seines and also in monolines. The rod and line.
Why do you think it's more common?
It's, you catch more fish at once, really.
Yeah, so if you think of the whole life cycle and cost of fishing boats, do you burn more fuel to catch one tuna fish with one boat, all per liter of tons that you collect? Or?
Yeah, I understand. I think my understanding in Norway is that they actually, so in 2022, they only used 50% of the purse seine quota because they were catching these fish in rod and line, actually. So they are finding that it's more efficient. I don't know exactly how, but I believe that it's because of the quality, because the quality of tuna that you get on rod and line is significantly economically more valuable, then it actually makes more sense to catch less fish at a high quality as opposed to catching many fish at a lower quality. Does that answer your question?
Well, but then I'm just thinking, what are the other big consumers of this fish? Japan, China, maybe. Do they prefer, because they do not use road lines, yes?
They do, to some extent, yeah.
If they want, so then comes to my next question, is this welfare research, is it driven by the industry, because they see an economic value, or is it driven by more NGOs through societal?
I think really both. I think that the fishermen, the industry, can really easily see the value of this. Because to them, it's money, right? Money makes the world go round. It's, for us as researchers, that's not really the goal, but it's like a really great benefit. So in my experience in Norway, it's very collaborative between the scientific team and the actual fishermen, even for tagging purposes. Like actually for this data, we had commercial fishermen that were out there with us that knew these methods, and if they caught a tuna, they would basically hand it over for tagging purposes. So I think it's both, really.
Okay, thank you.
Thanks.
Okay. Well, so congratulations. I like a lot your presentation because it has been clear, complete, and easy to follow for someone that is not an expert in the topic. So thank you for that. Well, just a few questions just to discuss a little bit and to understand better your work. About the methodology, I don't know, maybe you say that. How many fish did you analyze?
6.
Only 6?
Only 6.
Okay. Also, were doing the boat, preparing on the stuff, or just analyzing the...
I was not. This fishing happened in September of last year, so for us the thesis didn't start until January. I could not have been there.
No, I mean, just analyzing the recordings is...
Yeah, it was about over an hour of video footage that I looked at every second. So.
Yeah, so just to know and to understand better. And did you think that this model that you start developing in the close future could be used by the fishermen in Norway?
The model?
Yeah.
I think, yes, not at the stage it's at now. It needs, basically, I think the model could be really good if it has more data, but it's only 6 fish. It's also not the entire profile. It's not the whole timeline, because I have spots in each fish that I don't have any data from. So,
But after the entire developing?
I think yes, I think it could be really strong because I think we can make a very clear connection between
ODBA, which is really a proxy for energy expenditure, so physiological stress and behavior. And then the hope is for the future of this project that accelerometers can be used by the fishermen in real time to see the energy expenditure and to then have an idea of what is the stress state of this fish, which can be useful for a number of contexts, even in scientific programs, like where we tag and release fish. If we tag a fish that then dies straight away, it's quite a waste of that resource, of the time. So it's just one example of how I think it could be really helpful. Yeah.
Okay, that's all. Thank you very much and congratulations.
Thank you.
Thanks. So we still have time for one or two questions if there's any in the audience. I have one quick, oh yeah, maybe you go first then.
Yeah, I have a question. Do you also look at videos where they're not being fished?
Yeah, but the thing about bluefin tuna is that they're just like such a global species. They're so fast. I don't believe we have any videos of that. Like we do in sea pens and in cages and stuff, but not a fully wild observation. We don't really have those, not on video. At least not underwater.
My question was just going back to the understanding the welfare and the effect of what's happening. So Gunther was saying about them being quite quiet and either person, or you saying about them going back to the school and maybe not realizing their cost. How do we have any idea of quieter behavior or normal behavior actually being their stress response, and they are really stressed, but this is their response? Because some things play dead when they're super stressed.
Yeah. No, I don't think we really have the information. I think that in general, bluefin is fairly understudied because it's so hard to access, really. They're such a powerful, big, global fish. But that is very interesting. I think, though, when you look at the physiological measurements, that's when you can really see it's not stressful. Yeah.
So we have these data, the physiological measurements.
We have some preliminary physiological measurements, but not from capture events. Not from rod and line capture. We have it from other, tuna in sea cages and stuff.
Okay.
Yeah.
So
like that.
Put the pieces
Yeah.
together. Great. Okay, that's it. I think we have one more.
I heard you say that bluefin tuna being caught with purseine fisheries is less economically valuable.
If you look at it from a quality standpoint, it's just that in theory, I don't know the monetary numbers really, but I know that the less quality, the less price. And in a purseine, you have many more stressors in the capture process than you do in rod and line. Like crowding, there's a different type of gear injury, there's more of a risk of hypoxia as there's multiple fish in a small area. So that's just me basing it on stress, what I know about stress.
Okay, so all good, well done.
Okay, so we're moving to the second speaker in this session. I'd like to hand over to Virginia to introduce your master's thesis and provide us with an overview of what you carried out. So good luck.
Okay, thank you. Welcome everybody. I'm Virginia and I'm going to present my master thesis topic called Spatial Ecology of Kypelus riatus, Influence of Stegasis fuscus on on-range side in a coral reef ecosystem. So, reef ecosystems are a worldwide renowned hotspot of biodiversity. They sustain a wide range of wildlife and some biotic factors. They also have impacts in the reef ecosystems, such as herbivory and competition. In a constant challenge for space between coral and algae. And through these reef fish communities, they play a huge role into this dynamic, maintaining also an equilibrium between, again, algae and corals. And studying fish spatial ecology is not only important to know the ecology of the fish itself, but also helps understand the different players and how the ecosystem works, and also the interactions with the habitat. And pursuing this also further helps how to understand better the ecosystems and to push for more conservation efforts. In order to do that, spatial ecology, you can start by looking at the home range of the fish. And in my study, I looked at Chaetodon striatus, which is my species. It's a banded butterfly fish, only present in the Atlantic Ocean and from the Caribbean up to Brazil. And the other fish, whose relationship with the other one I'm also investigating, is Stegasis fuscus. It's a damsel fish present in Brazilian reefs, and it's also a keystone species. So it's important to say that no estimation on home range size for Chaetodon striatus has ever been done, and also the relationship between these two fishes was never really the topic of any research. So, as I said, like, the aim of my study is to characterize my Chaetodon striatus territories. Here in the picture you can see some of the territories, like 25 territories that we estimated. So in order to do that, I'm also looking at the interaction with Stegasis fuscus and the relationship with the environment. With Stegasis fuscus, there is an infer that they might be competing, so that's also what I was looking for. So my two main research questions are: what is the home range size of Chaetodon striatus, and which factors such as substrate coverage, feeding preferences, competitors' presence, and environmental parameters are influencing the area and extent of my fish. To do that, I went to Praia do Forno. It's a nice beach in Arraial do Cabo in Rio de Janeiro state in Brazil. And so this is where on the left shoreline we were sampling, we were doing our sampling efforts. And this is a shoreline sample, and it's long 700 meters. And it's where we also got our territories. Firstly, what we wanted to do was to get a full picture on the shoreline characterization, so what we found there. And so it's important to say that this area is characterized by low upwelling phenomenon, bringing a lot of nutrients, and it's also kind of an MPA, it's a marine extractive reserve, so it's not a no fishing zone, it's just mainly for livelihoods of the area. So what we did was mainly characterizing the area in two different ways. We did an underwater reef visual census, so we counted both fish species at the same time. We applied two different methods to be more safe and also to have a better view on the best method to use. And we used line transect, so we couldn't use the entire long transect because of GPS problem, we couldn't have done that. So we decided to do shorter transect, and of 50 meters each, we did 13. And in each transect, we were counting both fishes at the same time. We were also video recording, and we were counting both fishes at the same time on each side of the transect, the sand side and the reef side. And also we did a stationary point counts method. We counted fish in 14 points. 3 divers at the same time were counting the fishes for 3 times, repeating the counts. And in order to have 9 independent counts of the fishes, and we were of course looking in an area that we estimated to be 4 meters square. And also, I said that I wanted to look at the ecosystem and how the substrate was influencing, so we also did some benefit coverage analysis from the video transect that we had, and then I processed the 4 frames from the video transect in CoralNet, which is a software that does this, using 966 point for better precision. So afterwards, what we did was actually the main point of the thesis was to follow the fishes, video record them, track them, and we used GPS. We used a dive watch to follow the fishes, and we were also using the action cameras to, of course, video record them. From this, we followed the fishes for 20 minutes. The fishes were chosen randomly, but we still wanted to capture the entire shoreline, so we were choosing them randomly, but also moving throughout the shoreline to get not the same fish, so not to avoid resampling, but also get the entire picture of the entire shoreline. So what we got was the home range estimation from this data, because we followed the fish, and based on the track that the fish followed, we got some GPS points, and then on R I processed the data, also adding a themed buffer to adjust for the GPS sensitivity. See, these are some territory shapes of the fishes. And so we also got some behavioral data. I looked at the videos, we analyzed 18 minutes to get 2 minutes of acclimation of the fish behavior. And behaviors such as a number of attacks by Stegastus on Caetodo, a number of display occurrences, feeding, so how many bites, and also MaxM, so I also got a microabundance indicator. And also I got the fish size, because I used object of known length in the frame at the end of the following the fish, and I then analyzed it on ImageJ to get the fish size. And finally also environmental data such as temperature and max depth of the fish territory. And as I already explained before, I also did some substrate coverage on the fish territory themselves. So I analyzed the video and I got 5 frames, one every 5 minutes because it's 20 minutes. So I analyzed the 4 or 5 frames each territory to get a better picture also on the substrate of each fish territory. Here we can see some examples of attack. The main big one attack is going to come at the end. But yeah, we can already see some small ones. Of course, the Caetodon is the striped one, and the other one is Stegastus. So here it's going to be the big attack. Yeah. So I watched the videos, and this is an example of display. It's a smaller situation, there is no miting, and it's just flicking the tail. So here we go to some results. These are the ones from the characterization of the shoreline. So here we're showing the pie chart with the mean substrate coverage from the video transect. So this is the mean from all the transects, so the means, of 13 transects. We can see that the main substrate category we saw was Helicocaribeorum. It's a zoantid, it's clearly dominant also visibly in the reef, and it's covering most rocks. And then afterwards we also see some turfology, sand rock, and so on. And it's ecologically important to both species. And also this is showing the fish abundance data. I'm showing the mean fish abundance data from the stationary point method, and it was clearly consistent already in the water that Stegastus fuscus is clearly more dominant. And the dominance is so high that the Stegastus fuscus mean abundance is 5.12, and Caetodon striatus 1 from all the stationary point is 0.20, so it's like 25 times more Stegastus than my main fish. And we also tried some correlation between the two fishes' abundance, but it was not found to be significant. So the estimation of the area based on the 25 fish that I followed was, the mean area was 95 meters squared, and the range between the maximum and the minimum was from 40 to 205 meters squared. This is some characterization of the fishes that I follow, so based of course on the size, I could understand their moment in life, let's say. So the majority of the fish that I followed were adults, some others were juveniles, and some others were maturing adults, so they were around 12 centimeters, 12.4 centimeters is the size for majority, and they arrive up until 6 centimeters of length. Then this is like the location of the territory. I know that the majority of the territories that I sampled were in reefs, because that's where the majority of the fishes were living, and some less were in mixed situations, so in both reef and sandy bottoms, and the remaining ones were in sandy bottoms. And so the area was found to be smaller than Stegastus fuscus one, because that's already been studied, I have data on that, and also the shape is different, because Stegastus fuscus area is mainly circular, while Caetodon striatus is, we found out that it's mainly along the shoreline parallel and mainly vertical along the line. And here I'm also showing the occupancy, so I try to estimate, according to how many fishes, like the territory of the fishes, I know who I was following looking at the video, so I know that the majority of the fishes that I was following in the territories were living in couple, so they were mainly swimming in pairs and they were occupying the territory together. Some less were individuals, so just one fish per territory, and I also characterized like family situations, so like 2 fishes plus another one, so more than 2 fishes in one territory. And we got 2 occurrences of that, and it's normally 2 adults and a juvenile, but there is no parental care in this species, so it's mainly for territorial reasons. So these are the drivers that I also, the other results, and this is the mean substrate coverage from the territory videos, so 5 frames each of the 25 territories, and this is the mean of all of that. We see a similar situation, of course, to the shoreline characterization, where Halithoa is still the same, the main substrate category. This study, of course, confirmed also the competition between the two fishes. Stegastis is very aggressive. We got up to 22 attacks occurrences in 18 minutes video, and up to 5 display examples. And also here I'm showing, because normally in ecology, a bigger animal is occupying a bigger territory. So we wanted to see if this was happening also for our fish. Sadly, we did not find significant correlation for, between orange size and fish size, but we do see a kind of trend. So that's still something. So finally, to understand which factor actually influences the aerial extent of my fish, I got all the drivers, all the data that I collected, and we did some preliminary correlations, and that was the only variable that was actually correlated significantly and positively with orange size. But then we also did some model selection. We ultimately arrived to a best model, which is a general model with gather distribution and inverse linkage. And here is the best model. It was chosen according to having no residual errors and lowest AICC compared to the other models, and the ISR square, which is 40%. In behavioral studies, is not that bad, let's say. And so the first two variables, they're both saying the same thing. They're both different abundance methods for Stegastis fuscus, so the SPC coming from the stationary point data and the other one coming from the territory. And they're both negative estimates, so that means that there is an inverse relationship between orange size and the abundance of the fish. So higher abundance of Stegastis fuscus relates with smaller orange size of Chaetodon. They're both significant. In the model also, Halithoa made the cut, was positively present in the model, but was not significant, so we can't really say too much about that. So finally, the drivers of my fish orange. As I said, Stegastis fuscus abundance was the driver that was found to be significant. Not only maybe linking it to the size of the orange, but also we saw a trend about territory positioning, because most of the territories present in mixed and sandy situations were bigger, and also the bigger territories was actually registering a sandy bottom, and Stegastis does not go into sandy bottoms because its orange is very small, it likes to hide under rocks, so we may see future possibilities to explore this further, that maybe also territory positioning is dependent on Stegastis. This was also the case for another study by Randall on Chaetodon auriga, which is a Chaetodon species in Australia, and there was also negative correlation between orange size of this fish and Stegastis. And all the other drivers, variables did not significantly had an effect, but we did see some visible trend for fish body size, and this was also the case for Chaetodon trifascialis, another Chaetodon in again Australia, where gender and body size of the fish was correlated with membrane size, and for the others they were not significant, and also substrate coverage, as I said, it was not significant. This is the case for Chaetodon trifascialis too, but not for Chaetodon auriga, because in that study they actually found correlation between the percentage cover of sand and orange size for that fish at least. So as I said, this was the first study actually estimated the orange size of Chaetodon striatus, and also we understood that Chaetodon striatus orange size is negatively driven by Stegastis fuscus presence. The study actually highlighted the role of biotic interaction defining species ecology, spatial ecology, and also shedding light on the functional importance of these species, their role together, their role with the habitats, and their relationship all together. So I'm pushing for more behavioral ecology and species interaction studies, if it's not a new field, but like with a behavioral scape approach, we can explore something new. And to do in the future, we could compare multiple sites to bring more generalization to the study, because this is just one site and 25 individuals make the cut. and also research relationships with more species, not only Stegastes, because maybe they are also, you know, like just having other relationship with other fishes, or negative or positive. And also trying alternative methods to estimate homework size and software covers, such as KDE, MCP. We could not use that, so that could be something to be done in the future. These are my references. And yeah, thank you, and thank you to my two supervisors, Rafael and Tatiana. And yeah, thank you so much.
Very well done, Virginia. Thank you. So I pass to the jury members. Who'd like to go first? Lars, yeah, thank you.
Thank you. Your slides are very nice. Information inside that, but still possible to understand. I'm not a specialist of fish, and I was very surprised that Striatus lives about 100 square meters.
The territory that we found, yes, it's between 40 and 200, but the mean size of it's about 100.
Fish that size?
Yeah, it's maximum 16 centimeters.
So it's like very big.
Yeah.
It's so big because it needs a capacity of, because it's grazing algae on the current, so it needs a big surface.
Well, the species is actually an invertebrate. It's a non-coral generalist species, so it actually feeds on almost everything it finds around. I would say that in this area, that's why I would like to do it with maybe multiple sites, because in this area the presence of Stegastes is so high that maybe they're having this large territory because of the presence of Stegastes. Like Stegastes, it's dominant on all the shoreline. That means that they rely mainly on the sand, but in the sand bottom, they can only feed maybe on the trios. So they actually need to go around a lot to feed, or yeah, I don't know, just explore the area and leave. But that might be the reason. But also, I mean, compared to other species, Chaetodon ariga, it's slightly bigger, about, yeah, like 10 centimeters bigger, maximum. But its territory is actually up until 1,000 in some study, 1,000 meters square. So it's actually possible. And the other species that actually we compare it to is Chaetodon trifascialis, whose size, maximum size is 18, so they are actually in the same range. And its territory size is slightly smaller, so it's not that big of a difference. But I think it also really depends on like local balances and situationships. Like just, I guess that might be a reason.
And the competition for the fight between Fuscus and Striatus, is it related to the resource, or is it behavior which is more aggressive?
Yeah, at the beginning of the thesis, we actually wanted to understand this, but I feel like it's a very difficult objective to prove scientifically. So we do know that they feed on different things. Stegastes is mainly feeding on algae, and I mean Chaetodon feeds on everything. So they use both. Palitua is like ecologically important for both of them for refugia, because they like Chaetodon mainly refuge around, and Stegastes lives under rocks mainly. But since Palitua is cooperating with algae, that's like a garden situation for Stegastes. So actually they occur together. So it likes to protect this algae where it feeds on. So the competition is not linked to feeding, according to what we know, and it can imply from like papers on Stegastes and what we saw. But of course, it's not proven. And so I'm thinking that it's mainly for space, because Stegastes is actually attacking everybody, so that might be why. I think he's protecting his territory. It's not for, yeah.
What is the size of the territory of?
It's very small. It's like between 0 and 20 meters square. It's very small. Like there are several papers, slightly different estimation, but it's very small. It's very, like, it's just circling around. As you saw in the video, it's just going out if you see someone passing, and then it's just going back in. Also with people, it's very aggressive, so. Yeah. Thank you.
Thanks.
Yeah, thank you, very nice presentation, really enjoyed that. I wanted to come back to this absence of relationship between the abundances of the two species.
that's surprising. How do you explain that?
Well, I explain it with the fact that both methods of abundance, we got a lot of zeros. Because as I said, like Stegastis is very present, Kaiton is not. Even though we know that they are there, because we sample more than 25 territories with more exemplar living in each territory. But when we did both methods of abundance estimation, we got a lot of zeros. So I feel like when we did the correlation, we tried several methods, of course. We were looking for a triangular correlation, but we could not prove it significant. So it's actually like, if you see the points, there is a trend, but it's not clearly significant. So yeah, like the p-value that I'm showing here is very high. But I think it's mainly because, for example, for the lunch transect, we did not have time to do replicates, and that I feel like it's something, but we did not have time to do that. And yeah, mainly for that, and because there are not a lot of Kaiton striatus. If you just do a transect and you count the species, maybe it's there, but you can't count that. Yeah. And since we said that maybe Kaiton is not really present on the reef too much, then you count so many Stegastes, even though they are hiding under a rock, but there are so many that it's balancing the equation too much, and I feel like that's why it's not really significant. But there is something there, of course.
And I missed some information about the biodiversity context around this. I also think about Stegastes and Diptodon. I guess there's other fish.
Yeah.
Did you observe any interaction?
Yeah,
other
I
fish?
actually did.
How do you think they can maybe affect the relationship with this gene?
Yeah, I actually did. I mean, looking at the video, of course, you follow one fish for 20 minutes. I followed 29 fishes, so you have a lot of data that you can get from that. That's why at the end I put as possibility in the future to do multiple species too, because I actually observed some negative interaction also with other two species, Acantulus coreolus and another Stegastis pictus, I think. And so there actually might be something. It was not like I did not take note on how many times. I think it was like a couple of times, and it was not like bites or something, but still it was negative interactions. So that might be worth looking into. And plus, I actually would like to explore also maybe relationship with the conspecifics, because we got some videos of individuals, juveniles who were maturing adults, so not too big fishes. Maybe they didn't really found their territories yet. And while actually analyzing the video, the fish was like moving, just without really looking for anything, not really feeding too much. It was attacked by a lot of Stegastes. It was living alone, and when it was actually reaching or meeting randomly other conspecifics, they were not really treating it too much. Okay, so we observed some behavior of like tourbillon. So like you have fish, and the other one is going around it like that, making him lose like focus and balance. And it happened like twice. So maybe that's also worth looking into. And normally this happens when the other fishes, territory holder, maybe were more than 2 or 2. So it was also like 2 against 1 situation.
Okay, thank you very much.
Thank you.
Thank you. Well, congratulations for the report. Well, just a curiosity to discuss a little bit. One question about methodology. When you were recording the behavior of the fishes, you were there in situ?
Yes, I followed all the fishes. Not myself, of course. Yeah. Me and my supervisor, we followed all the fishes.
And do you think that that will have influence on the behavior of the fish?
Yes and no. We did some trial study, especially to understand if the 20 minutes observation period was enough to capture the full extent of the home range, because maybe it was not enough. Maybe the fish was, I don't know, just swimming too much, having a big home range. But we actually saw that within the 20 minutes, the fish was coming back to the starting point. So we thought that 20 minutes was enough, and it was also already done for Kaiton trifascialis. So we followed also that one. For the behavioral, I do believe not, because we still got the 2 minutes of acclimation that I did not count anything there, if not, of course, the track of the fish. But the fish itself did not seem very
startled or scared, not even interested. So I feel like it's a very different behavior between Chaetodon and Stegastus. Stegastus is aggressive, but also gets scared pretty easily. So like also when we were counting, just by passing the camera and counting the fish, Stegastus was either coming at you and then going back, or just going back inside the rock, while Chaetodon was pretty borderless, just unbothered. So I feel like we didn't really add too much impact on the fish. So yeah, that would be the reply.
Well, because I have checked some works like that, and normally they put the camera and wait for an hour.
Yeah, we also, like, early on in the planning of the thesis, we were planning also to do something like that, but then it was too much, because capturing the behavior, capturing the GPS, so you needed to follow the fish, and also how could you follow the fish, the same individual, because you can't really recognize these individuals. You don't know the sex, you can pick if the same individual that you recorded with the camera 2 days ago, or even just 10 minutes ago, is the same one that you're following. So that's the reasoning to do that was to get data on the same fish, and to do that you needed to do that at the same time. That's why at the end it was chosen to do it this way. But maybe something in the future to, like shoreline characterization, to do the same with like also the behavioral characterization for the entire fish population there, let's say. But then you can't really relate it too much, so that's why.
Well, just to finish, maybe it's a little bit out of the scope, but I am curious, so maybe I want to know your opinion. Well, your thesis is like the Striatus convergence is narrowly driven by Fuscus present. Do you think that due to, I don't know, maybe climate change, this relationship could change?
I don't know about climate change, but definitely there is something changing there, because I mean, based of course on what my supervisor has been telling me since he worked in this area in different projects for a lot of years, like at least I would say 10 years, since conservation efforts, especially really just linked to boats impacts, because there is mooring, but mainly just anchorage on the area. But these days is being protected way more and more each year. So linked to that, actually the substrate is flourishing the last few years, so it's getting better. The gorgonians are not bleached anymore, and but linked to all of that also the zoanthid, which is the main species of coral cover, let's say, there, is taking over a lot. And it's not invasive, but it's just very, very much abundant. And maybe that also actually it's something to look into, because I mean it's good for both species, but maybe it's also favoring Stegastus. So maybe it's like unbalancing also the equation. And Stegastus, it's aggressive with everybody, so it's not only about Chaetodon, it's also about like all the other fishes in the area. So maybe in the future, I don't know, but that was suggested by someone, removal of some cover of Bipanithua could be done. But yeah, climate change, I'm not really sure, because it's like a very peculiar area. We measure temperature, and it's like changing very much. Not in the day that I was there, in the 3 weeks that we sampled, but it's like, since it's on a quelling area, the temperature range is very big, like between 17 to 30.
Yes,
so
they are strong.
Yes, they are already very, I think, adapted at least to temperature, but I don't know the rest.
Okay, thank you very much.
Thank you.
Thanks a lot. Any questions in the audience here?
Hi, really lovely presentation. I was wondering how did the results of your study and the methodology that you used, how does that differ from, let's say, like habitat suitability model or like species distribution model?
I feel like it's tiny different, because there is some behavior behind it too. So we try to do all of it together. I'm sure that there are like better methods, as I say, for on-range estimation, and bringing actually species distribution model within this study would be, I think, bringing a lot of statistical significance to it and maybe more strong results. But the behavioral behind it, I feel like you can only do it this way and maybe not really using that kind of stats. So I guess that I really wanted to do something with behavior, not invasive. So yeah, that's why I'm, I guess, proposing that. Thanks.
Okay, well done, Virginia. Thanks very much.
to welcome our final speaker for this session and final speaker for today, so thank you for waiting until the very end. And I pass over to Rita to present your thesis. Thank you.
Okay, I hope you're not too tired. So my name is Rita, and today I'm going to bring to you the work I've been developed with my supervisors over the last 5 months. And yeah, I'm really happy to present this to you because it's in the topic that I've been very passionate about these last years. And yeah, I was lucky enough to find Miriam and Monica who were willing to guide me through this journey. But I must start to mention that this is not actually the title that I'm going to present to you, because today I'm going to present part of the results of my master thesis. And yeah, because we actually found like a larger data set, and we kind of, as we navigate to the data, we wanted to adapt a bit our plan. And instead of analyzing 3 species of baleen whales, we only analyzed 1, but in more depth. So I'm going to present instead the acoustic presence and behavior of fin whales during autumn and winter at a migratory habitat in the Azores archipelago. And to start, I want to give you a background of how fin whales acoustically behave. So fin whales are the 2nd largest species of whales, and as most of cetaceans, they take advantage that the sound propagates very well in the water, and they produced, in their case, low frequency sounds that go until 130 Hz, and these sounds can propagate thousands of kilometers in the ocean. Within this range, they have multiple types of vocalizations that we now know to have different biological functions. So they are known to produce certain types of calls in feeding context and other types of calls in reproductive context. So, taking the advantage of how vocal these animals are, we can use passive acoustic monitoring as a tool to study their presence and behavior at certain places. So, I'm going to start with the most well-studied call, which is the fin whale song. And these are basically stereotyped, repetitive, low-frequency sounds. And better than me using all these fancy words to describe it, I think it's good that you ever listen to it. So I'm going to play the sound, if it works. So, in 2002, there was a study that combined genetic data with acoustic data, and they found that these songs were only produced by male fin whales, which gave evidence that this could be related to reproductive displays, and also the fact that they produce more frequently these songs during breeding seasons. So, as you can see, these songs are very simple. So they are composed first, the most abundant call, it's the B note type, and the A note type. The B note type is centered on the 20 Hz, and the A note type between the 18 and 15. These songs, they kind of have some, although they are very simple, they kind of have some degree of variation. So they can vary in the proportions of the calls. Maybe some songs have more A notes than B notes, no, more B notes than A notes. And they can also vary in the peak frequencies that they use in the notes. But one very specific parameter that has been quite studied is the internote interval. This one is between B notes, and it's basically the interval between 2 consecutive notes. And this is going to be a very important concept throughout my presentation. So, as I said before, these songs, they have some degree of variation, and there was a very impactful study in this topic made by Action Mark in 2004. And what they described is that they combined genetic data also with acoustic data, and they discovered that the strongest determinant of difference among regional places, it was the internote interval. So since then, the interval has been used as to distinguish population, or at least we aim to. And so, however, like there's a growing body of evidence that this parameter, it has some limitations because songs are known to change over time within and between populations in both gradual and drastic trends. And so, other note that it's very common that females produce is the 40-year skull. And the 40-year skull, it's in higher ranges from 40 until 75 hertz. And these ones. And these notes, they have been positively linked with prey biomass. This study was specifically in the Azores. So they noticed that fin whales would produce more of these skulls when the zooplankton biomass was higher. And in the mid-Atlantic region, so these guys, they are highly migratory species. So they are known to go to high latitude areas to feed, and then they go down to low latitude areas where they are believed to breed. And in the Azores, the area is known to be like a stopover of these animals, so they are known to stop here and feed occasionally through satellite and visual data. But more recently, there was studies that described quite well the seasonality of fin whales, but with acoustic data. And these are the 3 studies that I'm going to base my study in. So, the seasonality of the songs, just to give you a background, the singing behavior, so the A&B call, tends to increase in autumn, reach the highest in winter, decrease in spring, and being absent in summer. For the 40-year skull, a different pattern was observed. So they tend to be low in autumn, increase in winter, reach the peak in spring, and also being absent during the summer. And so when we had new data, we had the opportunity to study a few research questions. So first, we aimed to compare the temporal acoustic presence of the fin whales in the new study period with what has been described before, to account for early changes. And then we decided to analyze the variation of the song patterns throughout the study period in a short scale, and then we incorporate specifically the measures of INIs in a large data set in a wide scale in the North Atlantic, and analyze the song trends of fin whales in this area and in this period. So, to achieve this, we use ocean-borne seismometer, one device. This device was located between 3 islands of the central group of the Azores, and was recording between August of 22 to January of 23. This data was provided by Upflow project, and after this, we were able, with this acoustic data, generating spectrograms by Raven Pro, which is the only software that we use. And with this, we create selection tables for the selections that we were, for the calls we were selecting in the spectrogram. And multiple parameters were used, such as the beginning and end time of the call, the peak frequencies, the invent power, so this was the energy per unit frequency per unit time, to study how intense was the sound. And this one is going to be very relevant, which is the time where 5% of the sound energy has already occurred. And all of these parameters are automatically measured by the software. Then, something that we also needed to establish is when we had a lot of overlap, so a lot of whales singing at the same time simultaneously. So, when these events occurred, we assigned a start and end time for these events, and we call them chorus events. And during these chorus, we couldn't select any call because there was so much overlap that it was completely impossible. Then, for song selection and measurements, the songs were selected manually, and we followed the same methods used in the previous study that we're going to compare, and a few assumptions were made. So, no masking of background noise could happen. The notes had to be quite distinguishable from the background noise, so they had to reach a signal noise ratio value of at least higher than 5 decibels. Then only one singer could be present, so no chorus, and other at least 24 hours of period between selected songs had to happen to avoid that we were selecting the same individual. And lastly, when we reach a resting period, because fin whales, when they are singing, they also take time to rest, you know. So when they take this time to rest, and this time was higher than 300 seconds, we would stop the selection. With this, we calculated the INIs, the average of peak frequencies, and the proportion of no times. And I'm going to emphasize as well that this was a very important parameter specifically to measure the INIs. So, what the results showed for the temporal and acoustic presence was pretty straightforward. So, for the song calls, the singing behavior reached a peak in mid-October. However, it's very important to take into account that this, the chorus events kind of creates the data here in this specific period of time. And since they reached the peak, they kind of stayed a bit high, relatively high compared to other seasons. And this was in agreement with what has been found before. Then, for the 40-Hz skull, they reached the peak in the beginning of December, which was quite different from what has been described before, because they tend to be higher as the winter advances, and for this period of time was higher in the beginning of winter, which also highlights the fact that there can be some yearly fluctuations in this culling behavior. So, we could conclude that fin whales, they have some kind of marked temporal patterns in the Azores, as has been described before, and that they are able to switch their calling behavior according to the season. So, although they can be present they can be present in a certain period of time, the fact that they are singing songs, they are producing songs, it doesn't mean that they are also not singing the 40-Hz skull, producing the 40-Hz skull, which yeah, it also supports what has been described before, that they have this ability of changing. So now for the variation of song parameters, we started to analyze the proportion of call types, and as we could see, not all the songs have the same proportion of A and B call. So a few songs they even only have B calls, which yeah, it's in concordance with what has been described to other areas in other studies. And the peak frequencies of A and B calls, they were highly correlated. So when one note has reached a higher frequency, the respective other note of the same song, they also had a higher frequency. Then, for the INIs, so to make this analysis, we plotted the average of INIs per song in function of the data was detected, and 40 songs were selected in total. What we can see, it's very clear that there is a high variability of INIs produced by the fin whales that use the study area in the study period. And something else that we could notice is that there was some kind of temporal pattern of the INIs used. So this we thought it was quite interesting. So in the beginning of the study period, the fin whales that were detected, they produced longer INIs compared to most of the fin whales detected towards the end of the study period. And then we plotted all these results and compared to the other regions in the North Atlantic and with a longer period of time. And so what we found is that this was probably the most outstanding results of the study, is that the INIs measured in the study period did not follow the same increasing and gradual trend of INIs. So in the study period, they happened quite an apparent shift in the INIs used, and they reset to shorter values, as the ones found 2 decades ago, which highlights the fact that songs are indeed, they have this plasticity capability, and it's also a limitation for the fact that I mentioned in the introduction that INIs can be used as acoustic markers for populations. So, and then all these things made me think like, why are these INIs changing? So we thought about some plausible drivers for these changes, and one of the first ones that we thought about is the fact that fin whales can be simply changing the INIs that they use when they reach a certain value. So they kind of could be that they have some physiological limits in the vocal apparatus, and they are forced to swift for shorter INIs. And this also has been found, for example, in this period of time and in other studies and for other regions as well. Secondly, we thought about like population mixing that can trigger vocal learning. This is something that has been described quite well for humpback whales, for example. They are known that each breeding season they kind of synchronize their songs among them, and it can happen that at some point the population of females that pass through to the Azores, they mixed with another females that use shorter INIs, and they kind of incorporated these INIs on their own songs. And later, we thought about there could be some kind of pressure that we were not aware of. So, naturally induced pressure that affects sound propagation. This was something that it can happen, like for example, changes in property of the water, such as oceanographic events, such as the North Atlantic Oscillation, or even the Kinis and El Ninos, which are events that affect this area of the Azores, or human-related pressures that create interference, such as vessel noise. And lastly, we decided to explore a bit further these changes in INIs, and we thought it was interesting to see how do they change. So what we thought was to plot INIs in function of the observations. And this was, when this graph appeared in my computer, it was very mind-blowing. So what it is here is that the females, they seem to produce songs in very stereotyped INIs values that differ among individuals. And it seems that they have constant ratios within these INIs. So, for example, we have this song produced by the individual, one individual in pink. The ratios are much smaller than, for example, the individual that produced the song 8. And this was quite new, and it was the first study that described these patterns. And of course, this has to be further explored, but there might be some information encoded in these patterns. And just to finish, I just want to leave you with a quote that I think very often when I'm trying to interpret it, give some interpretation to this acoustic data on communication of whales, which is a quote by this Austrian philosopher, which says that the limits of my language are the limits of my world. But language, of course, it's a bit polemic to use here, but if you think about it as communication, like the fact that it's so complicated for us to understand these animals, because like they have very different ways of perceiving the environment, of communicate, and yeah. So this is definitely a limitation for us to understand what information can be encoded and the drivers that change this vocal behavior in marine mammals. So yeah, with this I ended. Please, thank you very much to my supervisors and also for the project that provides the data for it. And of course, I want to thank the whales for being so chatty and giving me so much data.
Well done, Rita. Thank you. So I look to the jury for questions. Who wants to go first? Yeah, Esther.
Yeah, okay. Thank you very much for the presentation. Very nice topic. During the presentation, you have said that for the calculation of INI, it's very important a parameter that I think that is time 5%?
Yeah.
But you don't explain why.
Yeah, sorry. So we basically subtract this time to calculate the interval. So when the time 5% of one note, we subtracted two values of the both notes, and it gave us the interval.
And also, it is known is why the sound that you are measuring is only made by the males.
Sorry.
Why that sounds are only made by males?
So there's quite a few hypotheses. One of them is, for example, to attract the females to one spot. Others, they can kind of be competitive with the song, so they advertise their fitness to other males, and they kind of can be selective in that way.
But at the same time, this sound changes depending on the season.
Exactly.
So what?
Yeah, that's
It's
a good question.
100%.
That's the good question. Yeah.
Okay, that's all from my evaluation.
Thanks, Esther.
Yeah.
Yeah, Larissa, thank you.
Thank you very much. A very engaging presentation. At the beginning, you said that the sound at 150 hertz.
Sorry?
You said that the sound at the frequency of 150 hertz
120, yes.
propagates thousands of kilometers.
Yeah.
How does this long distance affect your results? How do you know that the way that you are listening to are the one with the Azor and not the other?
This is a great question. So, like, it's very difficult to account for distance when we are dealing with acoustic data. And there's a lot of factors that need to be taken into account. And I think when we want to study distance of the animals and density, we need also to have some studies in terms of the soundscape of the region. So have some kind of information about the topography, the properties of the water. So, like, when in the spectrogram, when the females were very, very far, when I saw that the girls were not like very intuitively selectable, I wouldn't count them because these animals would be very far from the region. So I had some kind of filters to select them.
And those filters were qualitative or quantitative?
They were quantitative when we did the song measurements, so they had to quantitative because I used the signal noise ratio, and they were kind of qualitative for the call rates. Yeah.
You have not how could you do it also in a more quantitative and objective way? I mean, there's the seal of 5% of noise to signature in the ratio is okay, but maybe you could also use some other techniques to classify this spectrum.
Like automatic detectors, like it would be a good way. We tried to use automatic detectors, but they didn't have the performance we were expected to. So we needed to adapt a bit, and it was mostly manual selection.
Okay. And then, why don't you show any results about the coverage situation? Isn't it an interesting situation where...
To be honest, because it's a complete mess. Like in the spectrogram, I cannot select anything. It's just a range of frequency that you can see that there are a lot of whales singing, but we cannot select anything. So for more meticulous measurements, this is not really reliable. What it could be done is that we can just account for the seasons that these events are more frequent, and
They could be more important during the breeding season.
Yeah, exactly.
So they need to breed.
Yeah, exactly.
And then, with with two songs, how many information can they achieve?
No, that's a question that I cannot answer. Unfortunately, I would love to have the answer for this question, but
how do they know that the distance between two waves, if the sound propagates so far?
Yeah, I think because the perception of our world is so like visual. For us, it's a bit abstract to just try to understand how can they perceive how long another individual can be. You know, this is definitely very like exciting research questions, but I don't have the answer.
How far did you think of the area that was been sampling just by listening? Was it 1,000 kilometers around the Azores, or was it 500?
No, because like the Azores also have, this is my guess, of course, my study was not relying on this
But
study.
it's an informed guess.
Yeah, so like, because the Azores have kind of like topographical profile. The topography is very like varying. So I think that whales that are very far wouldn't be detected because these sounds could reflect in like thousands of seamounts that there is around that area. Even like my study area, there was like it was in between two islands. So the fin whales that were singing like very close to other islands, in theory, I wouldn't expect to detect because like the sound would be reflected in the island and then travel in the opposite way.
So we know that sounds are reflected by seamounts.
Yeah.
Or they are just absorbed.
They are absorbed and reflected.
And are there some modeling of it?
There is a PhD student in our group working on it, soundscape. So maybe in the future we have some information about it.
Thank you.
You're welcome.
Yeah, maybe just a very small quick question. It was a very enthusiastic presentation. It's obvious that you love your whales.
I love the sound of my whales.
So it's kind of a follow-up on Lars's comments on the cores and also on the distance from which you can detect the wave. And can it be used to estimate the abundances of waves?
Yeah, like in the project that I'm working with, there are some people that are trying to find methods to account for density. But again, like it's still very in the beginning of the methodology development, so I don't have any answer like if it's possible or not possible. I think it would be somehow because like the fact that we can use multiple hydrophones, for example, like if we have 2 it's not possible, but if we have 3 we kind of can account for distance because with trigonometry we can calculate the distance. But then again there is the seamounts that reflect the sound and all of those kind of things like oceanographic features, and yeah, it's quite difficult, I guess.
Okay.
Thanks. Any audience questions? Oh yeah, yes.
Hey, I was curious if, because you said like you always left 24 hours between observations to make sure that they come from different individuals.
To avoid.
To avoid. Yeah. Is it so that is it sure that if
no, it's not sure. It's not sure. But like I could see that. Yeah, like when I would select a song, the other song, I could see that it was quite different, the patterns of the song. Yeah. But again, it can be the same animal, just singing different patterns.
Yeah.
But it was the only metric that we could use to try to account for different individuals.
Yeah. Okay. So at one of your last slides, you had different songs shown.
Yeah. These are definitely from different individuals, I guess, because yeah, the ratios are very different. I mean, yeah, it's quite difficult to tell.
To be figured out.
Yeah. It's very much in the air, like, you know, it's the first, when I saw this plot, I couldn't believe it was true, because I was like, no, this is too perfect, like, this is not even biological data, it can't be. But like, it is what it is, you know, and it was the Raven Pro who measured, it was not me, so there is some kind of information encoded here, and if it's through the individual level, there can have some potential.
Yeah. Makes a lot.
Excellent. Well done, Richard. Thanks a million. Okay, so that completes all of the sessions, but especially this session. So just one final thanks to all 3 speakers. Well done.